Are we back?
That's okay. That always happens.
Panelists. Are you panelists? Are you panelists?
Should I start sharing my screen again?
I'd say go ahead, Nicola. Thanks.
Okay, I will. Hang on one second. All right, are we back? Can you see that?
I can see it. Thanks, Nicola. Apologies, everyone, for the disruption. We'll crack on.
Hi.
Okay.
Sorry.
Sorry, Carolina, do you want me to go?
Yeah, keep it going. I'm sorry, I just got in.
No worries. So basically, I think what I was saying is that international livestock trade is occurring more frequently now than it ever has in human history, but we seem to have just sort of forgotten that it actually might be a vehicle for these parasites still. Parasitic diseases, because they are more they're less obvious than sort of the more overt viral and bacterial pathogens, often go unnoticed and generally aren't screened for before export, and definitely not upon arrival. Because there is a growing demand for a growing middle class in countries such as China and Vietnam, there is also an associated growing demand for animal-derived protein, which has outstripped local supply. And as a result, in 2019 in China alone, 150,000 animals per month were imported into the region, predominantly from countries where Fasciola hepatica is endemic, such as Australia, Argentina, and Brazil. And these animals don't just arrive at a port and then they're at their final destination. Many of them are being imported into various countries throughout the region and are being making their way up through various countries over transboundary trade. And this has sort of resulted in this sort of unofficial livestock corridor. And there's a great potential for the leakage of these systems and therefore the leakage of these animals and therefore their pathogens into these local systems. So for example, Laos is actually a significant livestock trade corridor into China. But in Laos, there's never been a recorded incidence of Fasciola hepatica, which is surprising given the number of animals that are moving through there, especially from countries like Australia, where it's endemic. So basically the hypothesis or the theory behind my PhD was that this international livestock trade is causing incursions of these parasites into the region, and there might be a more sort of insidious result of these parasite translocations events that are going undocumented. But to prove that, unfortunately, I needed to first develop a method that would enable me to screen animals ante-mortem and differentiate species that they were carrying, because up until that point, the only way you could confirm whether or not an animal was infected with Fasciola hepatica or Fasciola gigantica in a molecular way was by collecting the adult parasites from the liver at post-mortem during abattoir surveillance, which is first of all very low throughput, but also second, it's quite difficult to gain access to the animals at the abattoir. So it wasn't going to really work to use that traditional approach. The existing diagnostic methods at that time, including fecal egg counts and ELISAs, didn't enable species differentiation. So what I did was I had to challenge this dogma that you couldn't conduct species differentiation in a living animal, and that's because the material that you would need to access DNA from in these animals was the eggs, and the eggs were too robust for you to break open, especially compared to parasites that you would traditionally get in fecal samples. Very quickly, however, we disproved this dogma. You can see there's an image in the top right of the screen here, which is of a nice collection of Fasciola eggs that have been shattered open as a result of some robust beating. And this method that we developed was a very sensitive, high throughput way to isolate DNA from fecal samples. And we then compared it to the existing traditional diagnostic methods and found that, yes, the material that we're detecting is definitely the DNA that's coming from the eggs. This method is comparable to others in the point of first detection of infection, which in fasciola is about 8 to 12 weeks post-infection, because you have to wait for the parasite to do that migration through the body before it starts laying eggs that are excreted in the fecal samples. But we were pretty happy that this set a foundation for a plethora of different molecular species differentiation methods. So we in turn then developed a suite of molecular tools that included qPCR and traditional PCR, and also next generation sequencing that allowed us, depending on the locus that we targeted, to conduct a range of species differentiation and interrogation methods. It does have a limitation, however, and that is that if you are isolating DNA from many different eggs within one fecal sample, you actually can't differentiate an animal that has been infected with 2 different species at once and an animal that is infected with hybrids between Fasciola hepatica and Fasciola gigantica. But we'll get more on that later. For now, however, what I then subsequently did was go to northern Laos and collect samples from about 30 different villages in this livestock thoroughfare region. And I explicitly wanted to sample local animals that have been born and bred in the region, because if we were able to prove that Fasciola hepatica was in these animals, it meant that it wasn't just being trafficked through by animals going up into China, but actually the life cycle had established locally. And what we did was we found there was one animal we were able to detect pure Fasciola hepatica DNA in. We found a plethora of animals infected with Fasciola gigantica as expected, and we found a smattering of animals that were also infected with either sort of mixed DNA that we were able to detect. So they either had both species at once or had these hybrids. So the deal with Fasciola hybrids is that there is an increasing number of reports of hybrid or sort of introgressed forms in the literature. Many of these are coming from Southeast Asia. We were able to, using our molecular screening method, update existing maps of the distributions of Fasciola hepatica and Fasciola gigantica, and very stringently applied definitions for what we would consider Fasciola hybrid reports, and that's actually now available in John Dalton's second edition of the Fasciolosis textbook. But the problem that was happening is that people were reporting these events occurring, they were using these terms interchangeably, and there was no real consideration of what they meant, what was the functional outcome of hybridization or this potential introgression. We tried to set the record straight through the publication of an opinion about hybridization and introgression in Fasciola. We know that hybridization can occur. It has been done experimentally when you cross 2 parental species in a lab model, for example, but there's no robust evidence to support introgression. And I think it's because, for the most part, we're not looking at the problem in the right way, which is that we're not using a multi-loci approach. We're not looking at them at the genome level, but we do have tools available now that can support that. It's just a matter of time of actually applying them. But what's the deal? Like, what doesn't really matter if we're seeing hybridization and or introgression. The study where they were able to experimentally induce hybridization showed that the hybrid forms were not particularly fecund or long-lived, but because these parasites can exist in their host for a long time and be co-infected with multiple species at once, there is the potential that even just a single generation of hybrid offspring could generate back-crossing events between either of the parental species that would facilitate introgression of an advantageous allele into the genome of one of those parental species over time. And the concern is that potentially those alleles that could undergo introgression could have detrimental impacts on, for example, like the virulence of the parasites, their adaptation to a different host species, or even potentially drug resistance, which is a big deal. And the big deal is because with Fasciola, we have a very limited selection of drugs that are available to treat these parasites. Only one of them, triclobendazole, actually kills the immature forms with high efficacy before you start to see that significant liver pathology. It's also the only one that is registered for use in people, but it's the first one that resistance was developed against, and that was first reported in Australia, but it's now prevalent throughout Europe and much of the rest of the world. So there is, potential concern that if we're importing animals that are infected with Fasciola hepatica that carries resistance to some of these anthelmintics into the region, that there could be the transfer of resistance alleles into a space where not only is there potentially a large number of underreported human cases, but that over time you might start to find, diagnose, and then not be able to treat these cases because we're seeing the importation of either resistant parasites or introgression that has allowed for resistance to establish in a naive population. So that really means a lot, because these are the people here that have to sort of deal with this parasite. There's one health problem every day. It impacts their livelihoods in terms of economic losses in their livestock, but also it is a significant human health concern for them. So I think right now the focus is actually looking at the functional implications of hybridization and potentially discerning whether or not introgression is occurring, what traits are being introgressed into the region, and then also having a look at, is this a significant human health issue in other countries such as Laos, which is a major livestock thoroughfare in the region, not just in those neighboring countries where more attention is potentially being paid, such as Vietnam and Thailand, where we know Fasciola is hyper-endemic. And I guess that brings you to what we're doing here in Ireland. So we are part of the molecular parasitology lab here at the University of Galway under the leadership of Professor John Dalton, and he is currently running a Science Foundation Ireland funded project on the development of vaccines for future Fasciola control. Currently, the project we have underway at the moment is focusing on the development of vaccines for livestock, primarily sheep, but it is the hope that if we do get a vaccine that is effective in preventing infection or significantly reducing, for example, burden or egg output or impact of infection on the host, that would then be able to be later translated into a method of prevention that would be suitable for use in people. Regardless of whether or not we have human interventions as a result of these vaccine developments down the track, it would be a significant boost to our sustainable livestock parasite control, which would result in reduced importation of these parasites into the region, and also hopefully reduce the burden that's already occurring as a result of the ubiquity of these parasites in these systems that are so conducive to supporting this life cycle. So after all of that and a little technical glitch, I'm going to wrap up now, and I just want to say thank you very much for the opportunity to present, and I'm looking forward to any questions you might have at the end.
Thank you very much, Nicola. And again, we are so sorry for the glitch, and we cannot actually understand exactly what happened, but I'm happy to see everybody back. So let's continue. Nicola, thank you for keep going with your presentation, regardless the problems we saw there. It was a lovely overview on your experience and the problem that is Fasciola hepatica in the world. So let's move to our third speaker, which is Dr. Riona Walsh. I'm very pleased to introduce her and to have her amongst our speakers, because I think she will give us a vision on two problems that are not so much like maybe discussed. So Dr. Walsh will speak about One Health approach to tick and Lyme diseases in Ireland. And Dr. Walsh is currently undertaking her PhD in the ecology, epidemiology, and risk perceptions of Lyme disease in Ireland. She is a medical doctor by training and an assistant lecturer in health science at the Atlantic Technological University. And Dr. Walsh became interested by by disease ecology during a course of One Health that she completed at the University of Edinburgh, which led her to delving into the world of Lyme disease. She has special interest in the intersection between environmental protection and human health. So welcome, Dr. Rihanna Walsh, and thank you very much for your time here today.
Thank you so much for that introduction. I'll just start to share my slides. Now, can everyone see my first slide? Oh, great. Okay, I'm getting a thumbs up. Fantastic. Yep, so my talk will be on One Health as it applies to my specific research on ticks and Lyme disease in Ireland. My supervisors are Dr. Katrina Carlin is my primary supervisor, and Professor Mike Gormley of the Applied Ecology Unit in University of Galway. I realise now I should have changed to the new branding for University of Galway. I've also got external advisors in UCD and the Liverpool John Moores University, and those are Professor Annetta Zintel and Dr. Christopher Williams, respectively. And at different times, my research has been funded by the School of Natural Sciences Scholarship and the Irish Research Council. So during my talk, I will briefly talk about One Health as it applies to zoonoses and particularly disease ecology. And then I'll give you an introduction to Lyme disease and to ticks and ticks found in Ireland. And then I'll talk about my research in the ecology of Lyme disease and some of my data. When I think about One Health, the quote that comes to mind is this quote from the WHO, which is that many of the same microbes infect animals and humans as they share the ecosystem that they live in. Efforts by just one sector cannot prevent or eliminate the problem. And I do think that that rings so true for many diseases. We can try and approach it from one end and only to leave out an approach from a different end and find that really we need to always take a transdisciplinary approach that incorporates many silos. So that was the thinking as well behind kind of my approach to Lyme disease when Dr. Katrina Carlin and I started discussing kind of what the PhD would look like. And as we know, zoonoses are an infectious disease that has jumped to an animal, from an animal to a human, or is capable of jumping. And there's been many examples over the last 100 years of new and old zoonoses emerging or re-emerging, etc. So when I think of a zoonotic disease, I think of so many different moving parts. So we have the pathogen, of course, I always like to think of the pathogen, remind myself that it's an organism and that therefore occupies an ecological niche. So changes in the environment and in the different parts of the pathogen environment may affect the changes in the numbers of that pathogen, so the prevalence. Then there's, of course, the reservoir host. The host of the pathogen can infect, but may not necessarily get sick and then can pass the pathogen on. Sometimes a vector. So I have a mosquito here, but I look at ticks, of course, as the vectors, and then the end host. And then I think of the wider world of the environment around the disease and how perhaps changes in a habitat or the environment around it could change disease prevalence. And there was a paper that I come back to a lot that was published in Nature in 2020 that looked at 6,801 ecological assemblages, so many different host species and many different pathogens. And they found that in habitats that were disturbed by humans, so perhaps were degraded slightly, etc., or a lot, that there was loss of species from those habitats, but the species that persisted in the human degraded ecosystems were the same species that carry human diseases. So therefore, I suppose the conclusion you could take is that changes to land use impact human disease risk. So a lot of moving parts to think about. So applying all of those to Lyme disease, well, first I need to kind of give you a little background on Lyme disease. Many of you, I'm sure, have heard of it. So it's a bacterial illness caused by a complex of bacteria called Borrelia burgdorferi sensu lato, which is a spirochetal bacteria. Of course, as I mentioned, it's vector-borne, carried by ticks, and is the most common tick-borne disease in Europe. It's becoming prevalent in different areas that have seen Lyme disease before, and it's increasing in incidence in some areas also. Within Borrelia burgdorferi sensu-lato, there's about 20 different strains, but not all are pathogenic. In Ireland, we have Borrelia burgdorferi sensu stricto, so the nomenclature can be quite confusing sometimes. Borrelia guerinii, Borrelia velissiana, and Borrelia afzelii. And then, of course, the disease. So borreliosis is the infection with the pathogen, and Lyme disease, the symptoms. Stage 1 Lyme disease is the characteristic rash that you hear people talk about, the bullseye, the target lesion, and it's referred to as erythema migrans. And this is it here on me. So I was treated, and that was stage 1 done with. What you see is a red ring surrounded by an area of your usual skin tone, and then another red ring. If you see that, it's Lyme disease, it's treated with antibiotics. It emerges maybe days to weeks after bites, and sometimes is accompanied with flu-like symptoms. Stage 2 then occurs in about 10% of those who are untreated, and that's where you hear about the diverse range of very strained or sometimes very serious symptoms. Anything from arthritis to meningitis, affecting the muscles, affecting, causing nerve pain or palsies, and then there's neuroborreliosis. So I mentioned meningitis before, and it affects the nervous system. We refer to it as neuroborreliosis. And then stage 3 is that late disseminated phase that's chronic and often persists after the pathogen can even be detectable, with symptoms like arthritis, fibromyalgia, and fatigue. And of course, all this is caused by the bite of a tick. Ticks are the vectors of many diseases, not only Lyme disease, but also tick-borne encephalitis, which is a problem in mainland Europe. There's Rocky Mountain spotted fever, tick-borne relapsing fever, babesiosis, looping ill, anaplasmosis, the list goes on. This is how I identify the species of ticks I look at under the microscope. And this is Ixodes richness, which is the kind of tick in question for me, as is the main tick that would feed on humans who are walking through a forest habitat. As I mentioned, ticks tend, the ones I'm looking at, Ixodes richness, tend to favor woodland habitats, and they prefer habitat edges, so they congregate towards the edge of a habitat or transitional zones. They love high humidity. In fact, they would die in the absence of moisture, which is great for ticks in Ireland. Their development is inhibited by low temperatures. So people ask me, are they active in winter? And they tend to be much less active and more active during the summer months. They're found all over the world, but there's different species in different areas, and they're now being found in higher latitudes than before, and in fact, higher altitudes than before too, due to climate change. They are very undiscerning in their range of hosts. They can parasitize mammals, such as ourselves, or very small mammals, birds, and reptiles too. And they are exceptionally resilient. I've read papers that say they survive a wash, though they die in the dryer. And we also need to know how they become infected to understand the disease ecology of Lyme disease. So there's 4 life stages of Ixodes richness. They are born as eggs, hatch to larvae, take a blood meal, become a nymph, take another blood meal, become an adult, and then take a blood meal, reproduce. Again, hosts anything from small mammals, medium mammals, ourselves, deer, birds, reptiles. So when you think about it, the ticks that we need to be concerned about are the nymphs, because they've taken a blood meal, so they could be infected, and adults, because they've had 2 blood meals. Nymphs being more kind of threatening to humans because they're so small that it's possible that we wouldn't see them and remove them in time, as they tend to transmit the pathogen towards the end of their blood meal. And then the next thing I need to tell you all about is the dilution effect. This is a theory developed by looking at ecological assemblages in the USA that basically says that where there's quite a wider range of host diversity, the prevalence of Lyme disease in ticks is lower. However, that theory was developed in a place with a different tick species, a different primary strains of the pathogen, different host species, and vastly different ecosystems. The forests are much larger, etc. So it doesn't apply to Ireland. And just to explain how that effect works, I come back to these slides a lot. is we have a tick in a habitat with a range of different host options. And, you know, there's predators, there's small mammals, there's larger mammals. So the small mammals in this habitat that I've made up are the ones most likely to be able to carry the pathogen. So when the tick has a choice between different hosts, what there's about a 1 in 4 choice here that it will become infected. So some ticks infected, others not. In a degraded habitat, or a habitat with that's just less biodiverse, as I mentioned, degraded habitats are the ones where species that are more likely to carry human pathogens are more likely to persist. So we have more hosts that are more likely to infect a tick, and therefore more ticks become infected, and therefore a human passing through or living in that area is more likely to encounter an infected tick compared to the last habitat that we saw. And then we need to know about disease surveillance. So usually when we think of disease surveillance, we think of the human aspect. So reportable illnesses, you know, somebody gets a case of COVID-19, it's reported. In the case of Lyme disease in Ireland, we report cases of neuroborreliosis only, so specific symptoms occurring from the disease. In other countries, they might report all positive results, seropositive blood tests. And in some places, you even also report cases of erythema migrans, the skin rash that I showed you. You can also surveil the tick bites, and you can look also from a completely different angle. So that's the very base of the pyramid here, and look at the environmental aspects. So look at the questing ticks who are infected, and sample for infected wildlife as well. So I want to get a better sense of the base of that pyramid, which leads me to my research. So I'm looking at the disease ecology, the ticks, their habitats, their hosts, markers of disease risk to humans, what factors increase or decrease the prevalence in Lyme disease, does the dilution effect apply in Ireland? A bit on the epidemiology, though that data is very hard to get, particularly when you're working during COVID. And then I'm looking at the human side, so our perceptions on the disease and how that might affect our risk. So to get an idea of disease ecology, I've been, over the last 3 years, collecting ticks from various woodland habitats in Ireland to look at their density. I PCR them then for the prevalence of Borrelia burgdorferi within the ticks that I've collected from an area. In the same area, I'll also gather mammal data using camera trap footage and hair tubes, so they're non-invasive monitoring methods to get a sense of host activity in the area. Similarly, I'll take birdsong recordings and analyze those for bird activity in an area. Looking at the woodland habitat variables as well, the size, type of a habitat, vegetation structure, etc., and taking into account the weather, and all of that at the end of the day will go into a model which will hopefully help to model how habitats and hosts, etc., affect the nymphal infection prevalence, which is correlated with our risk of Lyme disease. And that's me actually doing it out in the habitat, dragging a towel, which seems very low tech, but it's how we sample for ticks. So the ticks sense my carbon dioxide and pheromones as a potential host, and attach to the towel thinking it is the host, and I pick them off and store them in ethanol. Secondly, I did a meta-analysis, so taking an approach which uses a tool often used in the medical literature and epidemiology, and applying it more to the side, the side of disease ecology. I looked at all the papers that have been published on ticks infected with Borrelia in Ireland, and then in Scotland as well, where we have a little more data on human risk, and compared the two as a proxy to compare human risk between the two countries. So this is the location of the various sites between Ireland and Scotland. And I looked at some environmental variables. So I looked at whether forest type might affect the nymphal infection prevalence. And I found that, in fact, coniferous woodlands in Ireland had a lower nymphal infection prevalence than mixed and deciduous ones, which was curious because they tend to have less host diversity in those areas. So that seemed unusual. Similarly, the forest size, as forests got bigger, the red line is Ireland and blue is Scotland. So in Ireland, as forests got bigger, there was a significant relationship with a higher nymphal infection prevalence, and there was a trend towards the same in Scotland, whereas studies from the US would say in larger forests, there's a lower nymphal infection prevalence. And this reverse trend is probably due to the fact that the woodlands in Ireland are very fragmented, and we tend to have very small forest patches compared to other areas in the world. In fact, that may also explain why in Ireland overall, our nymphal infection prevalence came out as 8.2%, which was higher than Scotland's at 1.7%. So if you looked specifically at the nymphal infection prevalence in Ireland versus Scotland, it was higher in Ireland, and they're otherwise quite similar places. So perhaps it has something to do with the forest fragmentation. So that gave a sense that if you looked at that only, that the risk of Lyme disease in Ireland is higher than in Scotland. And as I said, it's quite difficult to get epidemiology data for Ireland. We can look at the numbers of neuroborreliosis, and this is data from the Health Protection Surveillance Centre. So quite steady numbers over the years. But the problem is that Scotland doesn't record neuroborreliosis data, and it does record cases of Lyme borreliosis, but we don't. So the HPSC did approximate in a report in 2018 that there is maybe between 10 and 200 cases of Lyme borreliosis in humans per year, which would translate to 0.2 to 4 per 100,000 people per annum, which would be versus in 2019, for example, Scotland had an incidence of 5.64 per 100,000 people per annum. So a little lower in Ireland than in Scotland. So taking into account the NIP we talked about, there must be something in between. So perhaps it has something to do with human behaviour. So I'm looking at risk perceptions of Lyme disease in Ireland as well to get a sense of our behaviour around Lyme disease. If you looked at the literature around Lyme disease, you'll spot that people always talk about at-risk groups. So these are people who work in an outdoor setting or regularly use outdoor settings with high risk of Lyme disease, such as woodlands, for recreation purposes. So I've targeted both these groups via their workplaces or via recreation groups and did a survey on them. And I'm using the data from the survey right now to look at factors such as where we are in Ireland, our age, our hobbies, our professional background, etc., and how these may influence our perception of disease risk, to get a sense of how people think about Lyme disease and whether they feel they are at risk, what they do to mitigate that risk, and whether they've had training or are aware of the disease, can identify it should it occur, etc. The outcome I'm hoping to gain is to inform further training and campaigns in this area, and to learn how to inform people of risk mitigating behaviours. So in summary, I think I've covered a One Health approach to zoonoses and how I apply that within Lyme disease, my own research in the disease ecology of Lyme disease in Ireland, risk perceptions, and just as a final little tidbit, I would say, go out, have fun in the woods. When you come in, check yourself for ticks. Thank you very much.
Thank you very much, Dr. Rihanna. Was such a very nice, both scientific and medical perspective on the problems that ticks are in Ireland, and I think in the world also. I particularly have a few questions for you after. So I do have a reminder. I wanted to the audience just to pay attention to, because our Zoom meeting before just crashed, and I actually lost all the questions that the audience had put like before there. So if the audience can please put the questions again there in the Q&A, I really appreciate. So now again, let's move to our fourth speaker, which is Dr. Claire Kenny. We appreciate that Dr. Claire Kenny agreed to present in this conference, because I think COVID is being a little bit forgotten lately, and it is an important zoonosis, and we should be talking more about. So I'm very happy that she agreed to talk to us a little bit about her work, and she will speak about COVID-19 in Irish healthcare workers today. Dr. Claire Kenny is a specialist registered in infectious disease at the University of Galway Hospital, and she has experience in both biochemistry and microbiology also. Welcome, Dr. Kenny. Thank you.
Thank you so much. So I'm going to share my screen now. I feel like I'm bringing the buzzkill to the party because everybody does want to forget about COVID. I do agree. Is that working? Can everyone see?
It is.
Great.
Is perfect.
And thank you, Carolina, for inviting me to join in this very important conference. So let's talk about COVID in the context of it being a zoonotic pathogen, which of course it is. And of all these re-emerging infectious diseases affecting humans in this century, approximately 75% of them have been said to be zoonotic in origin. And when we think of that, we talk about HIV, we talk about Ebola and SARS virus, of course. And the zoonotic diseases that we are seeing emerging from our wildlife have significantly increased over the last 80 years. And really this last pandemic, I think it has brought to mind a number of concerns, including public health policy, how it integrates with our economy, societal concerns, and also environmental concerns in addition. So what about COVID? The current weight of evidence does suggest that the SARS-CoV-2 virus that causes COVID-19 or its progenitor probably emerged in humans from a zoonotic source back in 2019 in Wuhan in China. Now, the findings and the origins of SARS-CoV-2, well, they are inconclusive, but bats have been suggested to be the most probable evolutionary source for the virus. And when we align the full-length genome sequence of the virus, it shows the closest relationship with the bat SARS like coronavirus, BAT-CoV or ATG13. So an unknown intermediate host might have played a role in the emergence of SARS-CoV-2 in humans, and there has been a range of evidence in the literature, including intermediate hosts being pangolins, mink, rabbits, raccoon dogs, snakes. However, direct spillover of SARS-CoV-2 or its progenitor to humans from a bat host has also been proposed as a possibility. We do need further research to determine the exact SARS-CoV-2 evolution and transmission pathways. So why am I so concerned about COVID and preventable illness? Well, and why is the surveillance of our healthcare workers for these important zoonotic diseases important? Well, protecting healthcare workers from preventable illness and disability and death has to become a fundamental first step in building resilient healthcare systems that are capable of planning for and effectively responding to public health emergencies. And I think we'd all agree with that. And this is a picture, a lot of you are way too young to know what this is, but this is a canary and that's a coal mine worker. And healthcare workers, we do see that they really are the canary in the coal mine. What happens to the healthcare workers anticipates what will happen in the general community over the coming weeks. So when we look at Ebola and lessons learned, healthcare workers accounted for 25% of all infections in the 1995 outbreak in Kikwit in the Democratic Republic of Congo. However, later, when we go to about 10 years later, the Ebola outbreak in Western Africa, healthcare workers only represented about 4% of cases of Ebola virus disease at that time. So lessons were learned. And during the most recent 2014 to 2015 Western outbreak, a figure showed that the majority of transmission events were actually at that point between family members. So in many epidemics relating to the Ebola virus, healthcare workers have been the transmission link to the general population. And so really the importance of this healthcare worker transmission pathway appears to predominate in the early epidemic transmission chains and decreases as the epidemic evolves. So this is again saying our healthcare workers, well, they're the canary in the coal mine. And Ebola outbreaks really have occurred in areas which are resource challenged, which we all recognize that there are insufficient numbers of healthcare workers. They have reduced access and unfairly so to medical supplies and PPE. And what happens then, further reducing the number of available healthcare workers due to illness decreases resources available to provide treatment. And during the 2014 to 2016 Ebola outbreak, Well, there was an 8% decrease in healthcare workers in Liberia and a 23% decrease in healthcare services in Sierra Leone. So let's come back to COVID and our Irish healthcare workers, which is an area which I've been particularly researching over the last 18 months. And the breakthrough SARS-CoV-2 infections, well, they are of concern in the era of vaccinations, and we do remain a highly exposed health cohort. It affects staff well-being and quality of life. And again, absence due to illness and isolation leads to stresses on our already burdened healthcare service. And again, what we do see is that the healthcare worker cohort acts as a red light warning system to what is going to evolve epidemiologically in the community over the coming weeks. So we established, my colleague back in October 2020, Niamh Allen, the PRECISE studies, and they are looking at the prevalence of COVID-19 antibodies in Irish healthcare workers. And really at this stage, we're now launched our 5th phase today for our healthcare workers in Galway and in St. James's Hospital. So these are HPSC studies, and the 2 chosen sites are Galway and St. James's Hospital. So what do we do when we look at healthcare workers' antibodies? Well, really, I suppose what we do is we look at the anti-nucleocapsid antibody, and what that is that relates to your natural infection, a natural COVID-19 infection. If you've been vaccinated, what you'll see is an anti-spike antibody. So people who've had both natural infection and a vaccination will in fact be positive for both. Somebody who's been vaccinated alone and no natural infection will just be anti-spike antibody positive. So when we go back to October 2020, remember this is kind of 6 months on from when all the children were sent home initially from school. COVID had been diagnosed in 10.2% of James's healthcare workers and only 1.8% in GUH staff by the time of the study. And all staff at both hospitals were invited to participate in an online questionnaire and blood sampling for SARS-CoV-2 antibody, and there was a fantastic uptake rate. So there was seroprevalence of antibodies to SARS-CoV-2 was 15% and 4.1% in St. James's and Galway. And the interesting point about this is that 40% of infections were previously undiagnosed. And the risk of seropositivity for COVID-19 was higher in the people who do a lot of the heavy work in the hospital, including healthcare assistants and nurses. And of course, daily exposure to patients with COVID-19 was an increased risk. Living with other healthcare workers in particular, an Asian background and male sex. And in fact, the healthcare workers' seroprevalence was 6 times higher than was seen in the community seroprevalence at that time. So then 6 months on in April 2021, and at that point, our participants, our healthcare workers, for the most part, had been vaccinated. So we wanted to follow up and see what happened there. And really, seroprevalence to natural infection increased to 21% from 10.2% in St. James, and up nearly 8-fold in Galway. And that really does reflect, you know, the waves of COVID that had come through the population at that time, particularly that really difficult Christmas, January, February period of 2021. And what were the following risk factors significantly associated with seropositivity? Well, at that time, we looked at more social determinants, and Black ethnicity was an increased risk, a lower level of education. Again, living with other healthcare workers, being a healthcare assistant or a nurse in addition. And 26% of infections were previously undiagnosed. Now, interestingly, at that point of time in April, back in 2021, breakthrough infection occurred only in 0.6% of fully vaccinated participants. So it was a really low breakthrough of infection in fully vaccinated healthcare workers. We then went on to PRECISE 3, which really happened over last autumn, early winter, and finished up in January 2022. And what we did for this study is that we looked at all our PCR positive healthcare workers. They were invited to join a study, and we did a phone call with them at 0 and 14 days and sent their viral samples for whole genome sequencing. And what we did is we collected information on how sick the patients had become. And for example, all of these patients, participants, I should say, were vaccinated. In fact, 100% were vaccinated, so really remarkable uptake of the vaccine. And 94% of the people who had broken through their vaccine and become SARS-CoV-2 positive were living with cases under the age of 12. 86% were living with a positive case over the age of 12, and 81% were cohabiting with other healthcare workers as their main risk. And in the good news story, I suppose 29% of these people are asymptomatic. They are folks who were swab positive as part of a ward outbreak screen, for example. 71% were symptomatic. Only 3%, this is a remarkable contrast to earlier phases, required ED attention, and only 14% sought advice from their GP. And when we look at the breakdown of people's serology from that time, what we do see, and this is interesting, is that so 98% of people were positive for spike protein, but the people who broke through infections, only 2% of those had a previous infection, as in they were previously antinucleocapsid positive. So really this does support that vaccination coupled with infection really does provide a little bit of protection. So the low prevalence of baseline positive nucleocapsid serology in these breakthrough infections, again, I'm reiterating that infection coupled with vaccination may offer a more durable protection in this cohort. For PRECISE-4, this was in November 2021, and this is before we healthcare workers were just before we got our booster dose, our third dose. And at the time, the concern was the waning of immunity from primary COVID vaccination schedules. That's vaccine 1 and vaccine 2, and that really had been a growing concern. So we did sampling immediately before our booster doses, and therefore this provides an accurate assessment of the serostatus of this large healthcare worker cohort prior to the influence of additional dosing. So at that time, I suppose the total antibody seroprevalence was 23.4% in November, and that had increased from the initial phase of testing, PRECISE-1, of 10% in October. And this really does reflect the magnitude of the fourth wave in Ireland. And James's unsurprisingly had a higher seroprevalence rate at 24% in comparison to Galway at 21%. And obviously the incident infection rates were higher in the more densely populated Dublin County in comparison to counterparts in Galway. So in the October leg of the PRECISE study in 2020, healthcare workers in fact had a 6 times higher rate of infection than the general community, and this was less pronounced in November 2021, with SARS-CoV-2 prevalence estimated at 12.1% in the community, which is just under half of that seen in the PRECISE cohort. And so this finding really does demonstrate the increasing importance of community SARS-CoV-2 transmission as the pandemic has progressed. But yes, it does highlight that we still have a persistent increased healthcare work exposure risk. And in November 2021, 33.6% of infections were previously undiagnosed. And I suppose that really does drive home the fact of, despite evolving testing protocols, why do a significant number of our infections go undiagnosed? Is this through lack of testing or POSI or asymptomatic presentations? And the emergence of the Omicron variant very soon after study sampling, which was really about the middle of December 2021, further emphasized this issue, as asymptomatic infection rates were actually 32%. So now what next for PRECISE? So we are launching PRECISE-5, which is looking at a prospective cohort study over 3 years, and we're going to look at the evolving epidemiology of COVID-19 and the immunological response to vaccination. And what we will do is we'll transition from this episodic seroprevalence testing to establish a longitudinal cohort of healthcare workers. We will look at antibody responses and booster vaccinations. We'll also look at incident infections and diseases due to COVID-19, and T-cell immune responses will be performed in a subset. So in conclusion, healthcare workers do remain a highly exposed cohort, and I really do feel that enhanced surveillance of this cohort does remain a worthwhile process, as we do act as a red light warning system to what is going on epidemiologically in the community, and therefore we can prepare better and protect our healthcare workers better. I want to thank you. I don't have a nice photo of all my colleagues, but they were all very nice. And I want to particularly thank Professor Catherine Fleming, Professor Colin Bergen, Dr. Jonathan McGrath. And here are the references if you want to look back. But listen, thank you very much for letting me talk onwards. Thank you.
Thank you very much, Dr. Claire. Again, such an amazing talk. And I actually just moving then also to the Q&A and a little bit of the discussion, and I'll extend like a little bit the discussion, just 5 minutes, so then we can have a little bit like a 10 minutes discussion here. I worked with COVID and during the pandemic, and we did a lot of development of the diagnosis in the laboratory, and we did like a little bit of work with this, with samples from the PRECISE program, which is amazing. It's so well organized. It's so necessary. So
Thank you.
congratulations for your work. And I actually wonder, like, if you actually think that nowadays, like we are doing enough to protect the healthcare workers in terms of like testing, still, like when we have like the critical points of the pandemic, like testing and avoiding then to go to work when positive for COVID, or if that's is more or less normalized, like now with the progress and with the vaccination available and etc.
Yeah, I mean, I suppose really how PRECISE has transitioned is, you know, if you remember back to when we didn't have access to antigen tests, you know, I mean, really your access to getting a diagnosis was obtaining a PCR, waiting for the PCR to be performed. And I think people are much more cognizant now of when they have any coryzal symptoms that they do an antigen test and they stay home if they're positive. And that has really empowered the healthcare workers. But yes, I think, you know, the thing about antigen tests is, you know, we do still need healthcare workers, a certain proportion, you know, as we do in the community, of people to be PCR swabs so we can sequence, you know, the viral genome. And hopefully we are covering with, you know, vaccines that are coming out, and that's how they, you know, come up with different vaccine combinations with the new bivalent vaccines. So I think both. I think healthcare workers in general feel more empowered when they have antigen tests available to them. And I think from that point of view, yes, we do feel protected. We're always going to be more exposed.
Yeah, no, that's good. And is there any I know now that with the PRECISE 5 that you mentioned, like you are starting to also assess the antibody response to the vaccine on these healthcare workers, because I always wondered, like, we did see like a variation of how people respond to the vaccine and how long they take
exactly
to this immune response to wine, and then for us to vaccinate again and protect them when necessary, regardless the time that between the vaccines, especially with healthcare workers, I think.
So really what we want to do with the PRECISE 5 study is we're going to look at subsets of people, and rightly, like you said, some people like me, for example, I got my infection in January 2021, and at that time you were vaccinated 3 weeks later, you know. So people have different patterns of vaccination and infection and relating to the vaccine they got, the strain they had, when they had it, and when they were vaccinated. So people have a different imprinted response to COVID-19, and we are trying to elucidate what are these differences. You know, do we need a more tailored response over the coming years to vaccination? So that's really part of our next work with the PRECISE group.
Thank you very much.
Thank you so much.
No problem. I have a question for Dr. Rihanna Walsh that was put in here in the Q&A also. That's like it's an anonymous attendee. So the question is like how they thank you for your presentation and how they would check for a tick bite on darker skin tones. That was the question.
Okay, so when I check for tick bites, ticks, they could be 1 millimeter. So the ones that we're likely to miss can be 1 millimeter. And of course, on a darker skin, it's going to be even more difficult because the ticks are kind of dark in tone. What I do when I'm checking for ticks, I literally use the light from my mobile phone, and it's not even the color of the tick I see, it's the kind of raised area that I know wasn't there before. And then I'm able to remove it with the tweezers. If you're wondering about the rash, then the erythema migrans, yeah, unfortunately, a lot of the pictures you see, when you see the rash, it's always Caucasian skin, but it would still cause the rash with the clearing in different skin tones as well.
I do have a question, Dr. Rihanna. I saw that on your research you are collecting ticks from different areas in Ireland and also Scotland, and then you are PCR, you are doing the PCR to identify if they are infected and which bacteria is infecting them. Did you already identified some areas of concern in Ireland, like that would be more geographically speaking, more of concern, like forests or something like that? of ticks infected with important pathogens that would transmit Lyme disease?
I haven't, and the reason I'm focusing in on forests is because they have been identified as an important habitat for ticks in Europe. So I'm zoning in on the forest then to kind of tease out those factors. And the background that I'm working with is previous research done by people like Professor Gray and Professor Zintel, who have previously found that one area of concern is mucrus in Killarney National Park, which has a particularly high prevalence, and that prevalence does seem to be higher in forests than other areas as well. But that wasn't mine, that was somebody else's research.
I'm curious, like I'm sure will generate so much important data at the end of your PhD and your research. Thank you very much. Dr. Anthony O'Donoghue, if you were there, like I saw a question here for you, and I see you're interested on replying. I don't know if you are still there, or otherwise I will read just that Laura Gonzalez put a question here. Really interesting research and applications, maybe a basic question, but do the proteasomes act for removing fault or unnecessary proteins in the organisms? If so, how do you orientate your research to target higher all the proteins, say, from the parasite as target for the drug? That was her question. I cannot see. Oh, yeah.
I am here, yes. So it's a great question. And one of the ways we approach this is, rather than trying to eliminate single proteins, we're essentially trying to eliminate all proteins, or essentially by blocking the proteasome, all proteins within the parasite, or even within human cells when we do this for cancer, the proteins that have been ubiquitinated and have begun to be unfolded, they are super toxic to the cell. So by blocking the proteasome, we end up just creating a toxic environment inside either the human cell, this is for cancer, or in the parasite. And then the parasites themselves can't deal with this toxicity and undergo apoptosis. So we're not necessarily trying to prevent or increase the degradation of one or a few proteins. What we're trying to do is have a general buildup of undegraded proteins and leave the cell, and then that will actually trigger cell death pathways.
Okay, thank you. It's a very interesting approach, and I actually wondered the same thing. And also, I found it very fascinating your work with the plasmodium there, and like how effective is the inhibitor that at the end of the day you published there in Nature. I wonder how do you think how far we are like to progress to have actually an effective this transformed are made as an effective drug, like to be used and tested like in clinical trials, or I don't know if that progressed to this point already. I was just curious.
So the progress, so I think the malaria research that my group and collaborators are doing is we're getting close to having clinical candidates that maybe are getting closer to the clinic. One of the issues that we and my collaborators who have related molecules is that so far none of them have been very orally bioavailable, and for a disease like malaria it needs to be a single tablet, a single pill, and not an injectable. And so we are trying to, what we're really looking at now is the stability of these inhibitors in the stomach and the intestines, so that they can ultimately get survive the stomach, get absorbed into the blood system, and then ultimately kill the parasite in the blood. So that's what we're actually trying to do right now. And that's why the Gates Foundation and the Department of Defense have funded us recently and they want us to focus on making these inhibitors more orally bioavailable. Now, the reason I think it could get to the clinic sooner rather than later is that we now know that Novartis and several other companies have now taken an interest in developing proteasome inhibitors, GSK is another company, against parasites. So I think when you see the pharmaceutical companies seeing the types of research we're doing and saying, okay, there's something here, that kind of helps us, helps the programs move closer to the clinic.
That's interesting. And do you think that similar approach could be used for other parasites such as schistosoma? I saw that you working or other things.
Yeah.
So we lack drugs, like in general, drugs against parasites. So I'm just wondering.
Yeah, absolutely. Schistosoma is a parasite we're particularly interested here. The drug we have is not nearly as good for schistosoma as the one we have for Plasmodium. But there are groups, and actually GlaxoSmithKline have a molecule for Leishmania that's in, I think, phase 2 clinical trials now for Leishmania and Trypanosoma cruzi, which is Chagas disease. So there is some movement with other groups and companies to try and get these molecules into the clinic.
Do you think that the approach, like for protozoa, that are single cell parasites, are always easier because it's easier to have the effect of these inhibitors in the desirable cell, like, and that's with changes in parasites, in worm parasites, I mean.
Well, one nice thing about the parasites is they often go through these structural changes as they're working their way through the life cycle. And of course, those changes require a lot of removal of protein and building new protein, you know, as they grow through the phases. So that means they are particularly reliant on their translational machinery, which I don't target, and also on their degradation machinery. And so they are particularly susceptible to to ultimately dying if you can block that transition of removing old proteins so that they can make new proteins.
Thank you very much, Dr. Antonio. I really liked your research and your talk. Congratulations, and I hope to follow more your work from now on. To Dr. Nicola, just for us to change a little bit, I just have a question here for you, if you're there, Nicola. Yes.
Yes. Hi. Thanks, Carolina.
Okay. My question is, do you think hybridization is happening around the world, and which would be the areas we should be more concerned of that, like between the species of Phacelia hepatica and Phacelia gigantica, and which would be the areas, like on your experience, we should be concentrating maybe on using these molecular methods that you developed to investigate more the animals infected, like we can obtain the samples and eggs and etc. and perform this method?
Well, I think we can clearly see from the increasing number of reports of hybrid parasites in the literature that it is happening repeatedly. There do seem to be hotspots that are occurring, for example, in southern China and in the more focused on regions in Southeast Asia, such as Vietnam and Thailand. However, I think realistically what we should be doing is actually paying attention to any regions where we see live animal export. So, for example, we see a lot of live animal export into the Middle East, particularly, as the perspective of someone who's worked with livestock in Australia, that is a big market for us over there. And in fact, the first evidence that I was able to use as sort of foundation for my hypothesis actually came from abattoirs in the Middle East. So we definitely are seeing co-infected animals there, and where you see co-infections occurring and where you have the potential for live animals to sort of leak out of the livestock export chain is where you're more likely to see hybridization occurring. So I don't think there's any doubt that hybridization is happening. And I think that realistically we're pretty aware of where these sort of hybrid zones are, and they're certainly in regions such as the Middle East and Africa and Southeast Asia and southern China. But I think now collectively, while those reports are very important and they inform the magnitude of the problem, I think collectively we actually now as a scientific community need to be looking at what are the implications, what do these hybridization events mean. The molecular tools that I've developed certainly help for surveillance, but I think we also need to employ serological surveillance to be looking at the incidence of human fasciolosis in these regions. And then it is important for us to be looking at which species are causing human disease. So we can surveil using serological methods, but then we should actually be characterizing which species are causing human infection. It's generally presumed that Phacelia hepatica is more pathogenic, but that could be because there's a lack of actual data to support Phacelia gigantica, because people just not going to that level of detail when they're conducting clinical care. They sort of, one drug does treat both currently. Before you know, we end up with resistance in triclobendazole in human cases, which I think there is a little bit of debate about. But we do need to sort of stop before we get to just treat and move on and actually think, okay, which parasite is causing disease of concern in these regions, because that will inform information about the importance and function implications of hybridization occurring.
Thank you. And I will leave our conversation here today, like between us and me and other people and the panelists here, because then we give a break to the audience, which will be important because the next session that is on antimicrobial resistance is starting in about 10 minutes. So please, to the audience, do not leave. If you don't want, like you can always, of course, connect again. But if you want, just leave it on mute. And we'll be back in 10 minutes here with the antimicrobial resistance session. Okay, thank you very much again, Nicola, Anthony, Rihanna, and Claire. I really enjoyed the presentations, and I really appreciate your help with the conference and your sharing here with us. Okay, thank you. Bye-bye.
Okay, welcome back everyone. I hope you had a chance to have a little break before we move on. So I will be hosting the Focus on Antimicrobial Resistance session of the conference. My name is Dr. Georgios Miliotis, and I am a lecturer in bacteriology at the School of Medicine at the University of Galway. And I am very excited about the following session. So we have some very interesting talks lined up for you. So first of all, I would like to say that congratulations to the previous session. It was excellent. A lot of interesting talks, a lot of interesting questions as well. Okay, so in terms of antimicrobial resistance, we had Dr. Nitin Kumar Singh. He was supposed to give a keynote today, but due to unforeseen personal circumstances, he couldn't make it, and he extends his apologies. But instead, we're very excited to have Dr. Bridget Huben instead. So Bridget, she is a graduate of the medical science program at the Atlantic Technological University. She is specializing in medical microbiology and clinical chemistry. She completed her PhD in bacteriology at the University of Galway earlier this year, and her research involved examining a range of environmental sources for the presence of antibiotic resistance. Bridget now works as an assistant lecturer on the medical science program in the Atlantic Technological University, just across the road from us here. Bridget, you're very welcome. Very nice to see you.
Thanks very much, Georgios.
Okay, so the title of the talk today is Evaluation of Antibiotic-Resistant Enterobacterialis in Various Environment. And Bridget, the floor is yours.
Perfect. Thanks so much for that introduction, Georgios. So I'm just sharing my screen now. So it would be great if you could confirm that you can see it okay.
Yeah, we can, yeah.
Perfect. Thanks so much. So yeah, as Georgios said, I've recently completed my PhD at the University of Galway. So my talk today will be covering, I suppose, the work that I did as part of that PhD project. So the title of the talk is Evaluation of Antibiotic-Resistant Enterobacteriales in the Irish Environment. So just to begin with looking at the contents of this talk, so I'll begin with a very brief background and introduction on antibiotic resistance. Then I'll go through the overall aims of this PhD work. Then I'll get into some of the results that we found through our point prevalence survey. So this included our initial sample collection round across Ireland. Then I'll get into kind of some results that we generated through our longitudinal survey, which was 2 additional sample collection rounds across Ireland. And then I'll talk about, I suppose, some discussion points relating to the work and finish off then with some acknowledgements. So throughout the presentation, I've included some lovely images of the different, some sites that we were lucky to visit as part of the PhD work. So I hope you enjoy the images as well to break up the different sections. So some background information to begin with. So as we all know, antibiotic resistance is a global public health challenge. And in terms of, I suppose, the scale of the problem, back in 2019, it was estimated that 4.95 million deaths were associated with bacterial antibiotic resistance. And this was published in a recent article in the Lancet. So it's absolutely huge on the scale of things, especially from a worldwide perspective. So in terms of the actual pathogen drug resistance combinations that are responsible for these deaths, they're approximating between 50,000 to 100,000 deaths were caused by each of the following. So particularly E. coli is a very significant pathogen in the clinical environment, and particularly E. coli that displays resistance to fluoroquinolones, which are a type of antibiotic treatment, and also 3rd generation cephalosporin resistance. So 3rd generation cephalosporins are just a type of beta-lactam antibiotic. And also Klebsiella pneumonia that is carbapenem resistant and also 3rd generation cephalosporin resistant accounted for a significant number of these deaths. So in terms of CPE, which relates to carbapenemase producing Enterobacterales, these are, a very significant public health threat, particularly because they confer resistance to some of our last resort antibiotics that are available for treatment. So in 2017, they were actually declared a public health emergency here in Ireland. And just looking at some, I suppose, clinical data from 2020, we had approximately 694 new cases of individuals that were colonized or infected with CPE. So colonized essentially just means that they harbor this bacteria in their gut, but it's not causing an active infection. Whereas, of course, infected means that they might have, for example, an organism in their bloodstream or somewhere, some sterile site where it shouldn't necessarily be. So in terms of the types of CPE isolated then from kind of clinical samples, we can see that the species are listed in the left-hand side of the table, and the different types of carbapenemases are listed along the top. So the most common pathogen species that is associated with CPE or carbapenemase carriage is actually the Klebsiella species, followed by our E. coli and Enterobacter. Whereas when we look at the different types of carbapenemases, we can see that by far OXA48 is the most common type, accounting for 606 of the 694 cases. But we can also see some other types such as KPC, OXA181, and NDM are also relatively common here in Ireland. So in terms of, taking a One Health approach and kind of relating to I suppose, the overall conference here today, recent reports have recognized that antibiotic resistance is a One Health challenge. So we can't necessarily look at, clinical information in isolation, such as, human isolates or veterinary isolates. It is important that we look at, all three aspects that interlink. So the One Health concept recognizes that the health of humans, animals, and the environment that we all share are interlinked with one another. So there is an increasing need to better understand the role of the environment in particular in the spread or dissemination of antimicrobial resistance. So the diagram on the right here, you can see, I suppose, some examples of that interaction between the different components. So for example, if we look at our healthcare facilities, within the wastewater, we might have potentially antibiotic resistant organisms that are shed in our fecal material, which would subsequently travel to a wastewater treatment plant. But we may also have adverse weather events where our stormwater overflows are activated, and unfortunately that means that untreated wastewater would be discharged into our aquatic systems or our natural aquatic environment. We also have in Ireland some raw sewage discharges, unfortunately, that are discharging raw sewage untreated into the environment. And then how, I suppose, humans come into it is where, through recreational exposure, through actually drinking, for example, contaminated groundwater, these are all ways in which these resistant organisms can kind of come back full circle and interact, I suppose, with humans and animals. So following on next in terms of the actual aims and objectives of this research. So our primary aim was to examine both sewage sources and water bodies across Ireland and looking for the presence of different types of antibiotic resistant Enterobacterales. So we did collect samples across 3 counties, so Dublin, Cork and Galway. And samples that were collected then were examined for the presence of particularly ESBL and carbapenemase producers. So these are just 2 types of beta-lactamases that can confer resistance to different types of beta-lactam antibiotics. So Enterobacterales then were also compared at a genetic level to link both potential sources, so from sewage sources to resistant aquatic isolates. So we did find some interesting results when we did compare them, as we said, at a genetic level. So to get into looking at some results from our initial sampling collection round, which we call our point prevalence survey. And to note, I suppose, that this work was published in the Environment International Journal in 2021. So if you do have any interest, you can follow up with, I suppose, more information there, but you can absolutely contact me as well if you do have any questions. So initially, before we actually went out and collected our samples, we did create maps that plotted potential sources that may contribute to the environmental dissemination of antibiotic resistance. So this helped us choose both kind of hotspot sampling locations that were receiving, for example, multiple anthropogenic-based discharges, and also kind of cold spot locations which were, may have a low population in the area, might not necessarily have a lot of agricultural activity, or maybe free from discharges. So, that allows us, I suppose, to compare and contrast our findings then, and to help us identify potentially if the resistance that we're seeing in the environment could be from human origin. So in terms of these maps, you'll see that this map on the left is of Galway County and the map on the right is of Cork County. And we broke down the maps into electoral district level. So you'll see that the base colouring can be either red, yellow or green, and this simply relates to the livestock index. So areas highlighted in red would have, a high level of agricultural activity, particularly agricultural types that potentially may be receiving lots of antibiotics, such as kind of chickens and animals like that might receive antibiotics. Then we'd also have yellow to indicate medium agricultural activity, and green indicates low activity. And then you'll also see lots of individual points plotted on each of the maps. So these are the individual sources, such as hospitals, long-term care facilities, different types of discharges, for example. And we also looked at the actual water outside of the county as well. So for example, if we look at Cork here, we can see that the area around Cork City is highlighted yellow to indicate it's under significant anthropogenic pressures, whereas other regions would be highlighted red that potentially may have more of an agricultural based pressure. So again, all these maps and this work is published, and the link or the citation is listed there below. So for the point prevalence survey, once we did kick off our sample collection, we collected a total of 39 water samples and 25 sewage samples. So we collected them, and you can see this in table 3, across a range of different sources, including seawaters, rivers, lakes, drinking water treatment plants, as well as a range of sewage samples, including hospitals, nursing homes, airports, and wastewater treatment plants. And you can also see, I suppose, from these samples, we actually generated 211 individual Enterobacterales, or we isolated, I should say. And most commonly, we identified E. coli, followed by Klebsiella, Enterobacter, and then a few, I suppose, less commonly detected species. So interestingly, I suppose that we did detect the 2 species that are, very significant clinical pathogens. So in terms of characterizing these isolates, we performed antibiotic susceptibility testing, and we broke it down and compared the percentage of isolates displaying resistance to our panel of antibiotics into water versus sewage isolates. So you can see in general the percentage resistance seen for the likes of ampicillin, ceftibut-doxime, nalidixic acid was relatively high across both our water and sewage samples. So, the percentage resistance is along the outside of the actual circle. But interestingly, we did see a higher level of resistant isolates for water isolates in particular against the antibiotics ciprofloxacin and tetracycline. And tetracycline is actually the most commonly prescribed antibiotic for veterinary use in Ireland. So some, I suppose, interesting findings to see that higher level of resistance amongst water isolates. And then in contrast, amongst sewage isolates, we saw a higher level of resistance to cefoxidin and ertapenem. So these antibiotics would be, I suppose, more commonly used maybe in the clinical setting. So we also had a deeper look at the antibiotic resistance genes that were present in a number of isolates that we selected for sequencing. So by far the most common type of beta-lactamase resistance gene was our BLA-CTXM, which is an ESBL type beta-lactamase resistance gene. But we also found a number of carbapenemase genes, including our BLA-OXA48, BLA-NDM5, and also BLA-KPC2. So we also looked at, I suppose, other types of resistance genes that confer resistance to other types of antibiotic classes, in addition for our 60 sequenced isolates. So we see across the board, there's a range of different resistance genes present. There's, I suppose, no obvious difference between our water isolates, which are listed along here along the top, or our sewage isolates. So both harbor a very high number of different types of resistances. And you can see the resistance that the gene actually confers, the antibiotic class that the resistance gene confers resistance to. So we had a number of aminoglycoside resistance genes, sulfonamide, trimethoprim, and so on and so forth. So just to draw your attention in particular to something very interesting was that a number of isolates also harbored the phosphate resistance gene, which reduces the susceptibility of the phosphomycin antibiotic. So this is one of the antibiotics that's actually used for the treatment of CPE. So it is very, I suppose, worrying that we are seeing this, I suppose, so commonly, this type of resistance gene amongst environmental isolates. So to continue on then with the longitudinal survey, and this is also published online if you have any interest in reading further. So it was published this year in the Science of the Total Environment journal. And this involved 2 rounds of sample collection. So the sampling sites were chosen based off our kind of a results from our initial sampling round. And we tried to add on, I suppose, additional sampling locations where we did find something quite interesting. So for example, CPEs. So sample collection included 118 water samples and 36 sewage samples. And again, you can see the breakdown of the different sample types collected here. So the most common, again, was our seawaters, but we also included our other water types, as well as a number of sewage samples. And again, by far, we saw, E. coli being the most common pathogen that we isolated when we cultured our samples. But we also saw, similarly to the first round of collection, a lot of Klebsiella, Enterobacter, Citrobacter, etc. So in terms of the results then from the longitudinal survey, in terms of ESBL producers, we saw that 79% of waters and 50% of sewage samples harbored 1 or more ESBL producers. And a total of 18 water and 5 sewage samples harbored 1 or more carbapenemase producing Enterobacterales, or CPE. So if we look at figure 6 on the right, we can see that the bottom half of the image displays the actual sample types that the CPE were isolated from, and the top half of the figure illustrates the different types of carbapenemases that were detected. So for example, here, if we look at the yellow bar, we can see that in hospital sewage, we detected OXA48 and KPC2 producing organisms. So again, we've detected these across a range of both waters and sewage samples, but similarly to our clinical data, we see that OXA48 is the most commonly detected carbapenemase in the environment also. So interestingly, when we looked a little bit deeper at our sequencing data, we also found 4 isolates harbored the MCR9 gene, which is a gene that's associated with reducing susceptibility to colistin antibiotic, which again is very important antibiotic in terms of the treatment of CPE infections. And also the majority of sequenced isolates harbored 1 or more plasmids, so 140 out of 146 that were chosen for sequencing. And plasmids are really important mobile genetic elements that can essentially transport different genes from one bacteria to another. So it allows bacteria that capability to spread different types of antibiotic resistance in particular. So when we looked a little bit deeper or closer at these carbapenemase producers and looked at their antibiotic susceptibility profiles, you can see along the bottom is a list of the antibiotics, and the key for the shorthands is listed on the side. And then the center of the graph or the image illustrates whether the isolate displayed a susceptible, intermediate, or resistant phenotype to the individual antibiotics. So interestingly, when we grouped them by, whether they were an OX48 producer, an NDM, or a KPC producer, in general, we saw the OX48 producers don't harbor or didn't display as much resistance. And they also, the majority displayed in intermediate phenotype to our meropenem antibiotic, which is one of our carbapenem antibiotics. In contrast, then our NDM producers, we can see that they had a high level of resistance across, most of the beta-lactam antibiotics, and similarly for our KPC producers also. So it's quite a worrisome picture when we, actually have isolated these organisms from the environment, and some of them only showing, for example, these two here, susceptibility to one antibiotic. So if those isolates were to cause an infection in a human, they would be very difficult to, treat successfully using antibiotics. So the final type of results that I wanted to share with you today as well is, as we said, we looked at comparing isolates at a genetic level across different sample types, and in particular to try and, I suppose, identify if there were particular isolates in the water that could be linked back to sewage sources. So in terms of our E. coli comparisons, we performed core genome MLST typing, where essentially we look at a number of different loci and compare it across different bacterial species or different bacterial isolates. So within these clusters, we saw that there were isolates from seawater and a wastewater treatment plant influent in Cork that actually had 100% allele similarity. So this would indicate that they are, very, very similar isolates, if not identical. And again, potentially the isolate in the water may have originated from human wastewater. However, I suppose just to mention here, we didn't detect this isolate in the wastewater treatment plant effluent. So although we did collect a sample of the effluent, we did not detect it there. So we can't say for certainty that it did originate from the wastewater treatment plant. So for cluster 2, then we saw an identical isolate from a nursing home sewage and an estuary in Galway. And these were located 14.5 kilometers apart, but they did show 100% allele similarity. So again, potentially showing us that the isolates in the waters may have originated from human sewage. We also looked at our Klebsiella isolates and compared those at a genetic level. Cluster 1 and 2 are isolates from different water sources around Cork, which again showed a high level of similarity. And very interestingly, I suppose, for cluster 1 is that these isolates spanned a 9.8 kilometer distance. So showing us that potentially some organisms might persist in the environment and, be able to disseminate these types of resistances across a large distance. We also saw then for cluster 3 that there was isolates from actually a river in Cork, a wastewater treatment plant influent, and estuarine water in Galway. So there was a large distance between the isolates obtained in Galway versus those obtained in Cork. So again, very interesting results to see that similarity on such a wide scale. So to finish then with some points of discussion. So we did see a repeated detection of CPE in the aquatic environment, and particularly across urban areas. So I haven't been able to go into too much detail today for the results, but for the samples that we did collect from kind of cold spot locations, we didn't find any indication of carbapenem resistance or carbapenemase producers in these areas. So this would indicate that there is a potential anthropogenic origin or human origin for these isolates in the environment. And similar resistance types are circulating in both the clinical and natural environment. So again, showing us that link between human health and the environment. So we do have currently knowledge gaps relating to colonization of antibiotic-resistant organisms caused by exposure via the aquatic environment. So what this means essentially is that individuals that may participate in recreational activities and actually ingest some of the water, how likely are they to actually become colonized with these antibiotic-resistant organisms or get them into their actual gut? So there is ongoing work as part of the PEER project that you'll hear about later that is investigating this further. In terms of our CPE detection, it was very interesting that we found them in hospital sewage, wastewater treatment plant influent, and airport sewage. And this, I suppose, brings up the question as to whether we should be surveillancing, for example, our hospital sewage or airport sewage for things like emerging threats. So there are some countries that have trialed, you know, a surveillance technique where they look at the sewage coming from hospitals or wastewater treatment plants influent to kind of help establish the numbers of individuals that might be colonized with a particular organism. Also, on-site treatment is another kind of interesting aspect to consider. Should we be treating, for example, hospital sewage before it actually enters into the main sewage network to prevent that kind of spread and dissemination of resistance genes across bacterial isolates? Our core genome MLST comparisons reveal genetically similar isolates across both water and sewage sources. However, there's a lot, I suppose, unknown in terms of the gene transfer mechanism amongst aquatic isolates. So in particular, when we did see isolates across a large distance that were genetically identical, it's interesting to consider how these organisms can kind of persist in the environment and how they actually spread different resistance genes or plasmids that we might have seen across them. Also important to note that in general, I do think that Ireland's wastewater management system does require intervention. So throughout the duration of sample collection for this project between 2018 and 2020, the number of raw sewage discharges only decreased from 36 down to 34. So there's a lot of work that needs to be done to actually eliminate any types of raw sewage discharges into our natural environment. Also in 2020, 12 wastewater treatment plants were actually non-compliant with the EU treatment standards, and also 27 discharge events occurred during the bathing water season alone in 2020. So the bathing water season is the months between May and September. And in this case, that means that raw sewage that was untreated was being discharged into our natural waters. So again, we need to, I suppose, look at capacity of treatment plants, potentially building more treatment plants and things like that, and having better systems really if there is adverse weather events to prevent any kind of leakage from the network. And in general, there is limited monitoring of the aquatic environment. So we don't have any routine screening for antibiotic resistance, and we do need a harmonized approach. So it brings up again an interesting question that there's no kind of ideal marker defined. However, the World Health Organization did publish a tricycle protocol where they suggested people look at ESBL E. coli to monitor kind of human, animals, and the environment. So to kind of essentially have some marker that can be comparable across the three. And just to finish then on some future work that's kind of ongoing as part of the ARREST project. So there is work ongoing looking at environmental CP isolates and comparing those to actual clinical isolates to establish, again, if we can link back the isolates found in the environment to potential human origin. We're also undergoing further investigation of the MCR harboring isolates and whether they actually confer resistance to colistin. There's also a risk ranking analysis paper that is being drafted that is looking at, I suppose, the risk of environmental exposure to these particular organisms. There's also a shotgun metagenomic study that's looking at essentially the entire DNA content in our particular water source. And then point 5 and 6, just to finish up quickly, are more generalized points that need kind of future work. So kind of a more standardized methodology for routine surveillance of environmental antibiotic resistance. And also the nexus or connection between recreational environmental exposure and colonization with resistant bacteria is also needed. So thank you very much, guys, for your time and attention. Just to acknowledge everybody that was involved in this work. Definitely couldn't have done it by myself. So thank you very much, and I look forward to answering any questions later.
Thank you very much, Bridget. Fantastic. A lot of work. And I certainly appreciate all the serene pictures from the Irish coastal line, where I'm sure you have other pictures that were not as serene when you were collecting samples in the winter.
Absolutely, yeah. When it was freezing, it wasn't as nice to be out collecting water.
Anyhow, we'll come back. There are some questions already for you, but we'll come back during the Q&A session. So thank you so much for now.
Thank you so much.
Thank you. Okay, so we're going to move on. And so for the next talk, we have Francesca McDonough. Francesca is a research assistant in the Antimicrobial Resistance and Microbial Ecology group at the School of Medicine at the University of Galway. Francesca has an undergraduate degree in neuroscience from University College Dublin and a master's degree in biotechnology from the University of Galway. And her work encompasses microbiology, neuroscience, and genomics, and focuses on the link between mental health disorders, antipsychotic usage, and infectious disease. So we're very much looking forward to this talk, Francesca. The title of the talk is The Missing Link Between Antipsychotic Use and Multi-Drug Resistant Pathobionts. So ready when you are, Francesca.
Thank you.
We can see that fine. So
Yep.
ready to go.
Grand. Yep, so my name is Francesca and today I'll be giving you a talk on the missing link between antipsychotic use and multi-drug resistant pathobionts. So this work was done as part of my master's in biotechnology project, and this work is part of the Beacon study, which looks to shed light on the link between infectious disease and schizophrenia. So first I'll give you an introduction into what schizophrenia is. So schizophrenia is a multifactorial disorder that affects approximately 0.5 to 1% of the global population. The age of onset is in young adults, so around the early 20s. And of all mental illnesses, patients with schizophrenia are known to be the most commonly hospitalized with infectious diseases. It was estimated that the cost of treating schizophrenia was 29 billion in Europe in 2010. And patients with schizophrenia have a reduced quality of life and also have a reduced life expectancy by 5 to 10 years. So the symptomatology of schizophrenia can be divided into 3 main categories. So you have positive symptoms, which includes hallucinations, negative symptoms, which include social withdrawal, and then cognitive impairments. Approximately 30% of patients that are diagnosed with schizophrenia are diagnosed with treatment-resistant schizophrenia. So this is the persistent of schizophrenic symptoms despite at least 2 appropriate trials of different antipsychotics. And the first line and really the only treatment for treatment-resistant schizophrenia is clozapine. So then what is clozapine? So clozapine is an atypical antipsychotic, and it acts as a selective antagonist at the serotonin 2A receptors and the dopamine D2 receptors. However, it has antagonistic effects on a lot of other auxiliary targets. And it's this combination of targets that's thought to partially explain why clozapine is the only antipsychotic that does work to treat treatment-resistant schizophrenia. However, a main limitation of clozapine treatment is its side effect profile. The most notable side effect would be neutropenia, which is a reduction in the number of white blood cells. And this can lead to an increased risk of infection. So in Ireland, when you start on clozapine treatment, you have to undergo weekly blood monitoring from week 1 to 18, twice weekly blood monitoring from week 18 to 52, and then monthly blood monitoring from week 52 onwards. This risk of clozapine-associated neutropenia has also led to issues with the treatment of infection for people who are on clozapine. So there are 3 main contradictions between antibiotics and clozapine. So these are quinolones, in particular ciprofloxacin, macrolides, in particular erythromycin, and trimethoprim and cotrimoxazole. So ciprofloxacin has been known to inhibit members of the cytochrome P450 system, which can result in an increased serum concentration, and this has known to be potentially fatal. Erythromycin also can inhibit a member of the cytochrome P450 system and can induce neutropenia through the increase of serum concentration of clozapine. Trimethoprim and cotrimoxazole, however, operate in a different manner. They have been known to increase the risk of a blood marrow suppressive effect, which can go on to lead to neutropenia. There's other antibiotics as well that are associated with an increased risk for clozapine-associated neutropenia, and these include benzodiazepines, sulfonamides, cephalosporins, and clindamycin. So if possible, these antibiotics are avoided if treating an infection in patients that are on clozapine. So this project looked at samples from the oral microbiome. So the oral microbiome is very diverse and unique, with more than 1,000 bacterial species identified so far. However, it is relatively unexplored when compared to the gut microbiome. So Enterobacterales are not typically part of the human oral microbiome database, and their presence in the oral cavity has been linked to oral and systemic diseases ranging from HIV to cystic fibrosis. So the main hypothesis of this work was that patients that were on antipsychotic medication, specifically clozapine, will have a high prevalence of oral colonization with opportunistic pathogens that are known to cause pulmonary infections. So the methodology that was employed, patients with a diagnosis of treatment-resistant schizophrenia and therefore on clozapine were recruited from University Hospital Galway. 2 milliliters of unstimulated saliva was collected from each patient following informed consent, and then these samples were brought back to the lab for same-day processing. So these saliva samples were then screened on selective agars for the enumeration of Enterobacterales and also to detect ESBL producers, carbapenemase producers, and colistin-resistant organisms. So isolates that were obtained from these saliva samples then underwent antimicrobial susceptibility testing, and DNA was also extracted from these isolates to undergo whole genome sequencing, both short reads by means of Illumina and then long reads by means of Nanopore. And then bioinformatics processing was carried out on this genomic data. Metadata analysis was also carried out by collection of the patient's medical history after their consent was given in order to determine if there was any additional risk factors such as previous infections or hospitalization that would contribute to the oral colonization rate that was identified. So first, I'll just talk about the clozapine cohort that was recruited. So these are the inclusion and the exclusion criteria. Of particular note is that these patients could not have used antibiotics in the previous 2 months before they provided a sample. So these are the demographics. So we managed to collect or recruit 45 patients to the study. We had a wide age range from 25 to 81. So these were the different demographics: gender, smoking, living situation, use of antidepressants, the dose of clozapine that these patients were on, and their white blood cell count. We determined that none of these demographics had a contributing factor to the colonization rate that we observed, and therefore were not risk factors. Of the 45 patients that we did recruit, 23 or 51% were identified to be orally colonized with Enterobacterales. So I'll just go into a bit more detail about this. Of the 23 patients that were colonized with Enterobacterales, we identified 32 different isolates, which were clustered into 13 different species, which you can see in the plot on the right. 10 patients, or 43%, were identified to be multi-colonized, and 6 isolates were identified to be multi-drug resistant. So the most prominent species that we identified was Klebsiella oxytoca, followed by Serratia, and then Panthea. We conducted antimicrobial susceptibility testing on all the isolates. So the red indicates that the strains were resistant, and then the green is sensitive, and then the blue is intermediate. So of all the antibiotics that we did include in the antimicrobial susceptibility testing, only 4 displayed no resistance. So that includes kanamycin, ertapenem, meropenem, and cephalexin. And then ampicillin displayed the highest resistance amongst all the strains. Using the genomic data, then we were able to identify antimicrobial resistance genes that were present within all the isolates we identified from the cohort. So that is just depicted here. So the ARGs are listed on the top, very top of the diagram, and then on the right-hand side, the strain numbers are listed. We also used the genomic data to identify virulence factors that were present within the genomic data of these isolates. So the ones highlighted at the very bottom of the graph were identified to be hypervirulent. So that was 15 out of the 32 total isolates, or 46%. We also looked at the average nucleotide identity for the isolates. So average nucleotide identity, it looks at genomic similarity at a nucleotide level in the coding regions between different genomes. So a cutoff score of over 95% indicates that 2 isolates are from the same species, and a cutoff score of over 99% indicates that 2 strains are from the same strain. So we'll just look at 2 clusters of interest. So strain 28 was identified to be Calamella personii, which I will talk about later on in the presentation. And isolate 69 was determined to be a novel species of Calamella. In this cluster, strain 61, 62, and 63 were determined to all be strains, the same strain of a hypervirulent Citrobacter cocceri of ST854. And of note, these 3 strains were collected from 2 patients who came to the clinic on the same day. So I'll just talk a little bit more about Calamiella personii. So the strain of Calamiella personii that we identified from the study here in Galway is referred to as GABE KP28. And Calamiella personii was first identified on board the International Space Station in 2019. So this is a visualization of the genome of GABE KP28. And on the inside in the orange is a genomic comparison to different strains of Calamiella personii. So the 4 on the outside are strains that were identified from the International Space Station. And the strain on the inside is a strain that was identified in Mangalore in India in a case of kidney stone disease. So the colors then on the outside, the very, very outside ring, the colored rings, they are the genes, and the genes then are colored according to their function. So as you can see, our genome that we identified in Galway was quite similar to the ISS strains. The strain that we identified in Galway had 3 mobilizable plasmids. However, of note would be plasmid 2. This is a large hypervirulent plasmid with a suspected susceptibility type of INC-F, and this plasmid hadn't been seen before. So I'll just go through a few areas of note in this plasmid. First would be the EMRA and EMRB genes, which are multi-drug efflux pumps. These pumps are associated with colistin resistance. However, this plasmid, it does have a reduced susceptibility to colistin, but it is not resistant. This is the vibrobactin gene, and then this encodes for the aerobactin operon. So they are 2 major virulence factors. And the VIR-B gene regulator counters a transcriptional silencing of virulence factors. So therefore it allows for the expression of virulence factors. So altogether, this is quite a hypervirulent plasmid. And in addition to being resistant to drugs, GABE KP28 confers resistance to heavy metals, disinfectants, and UV light. So just in conclusion, patients on clozapine were identified to be more likely to be colonized with drug-resistant pathobions. No other factors from the ones that we recorded in this project appear to affect or influence the colonization rate. 45% of the strains appear to be hypervirulent, while 17% of the strains appear to be multi-drug resistant. And in terms of future work, we aim to recruit a larger cohort of patients and to include metagenomics and neuromicrobiological analysis. Thank you for listening.
Francesca, thank you so much. Great talk. Thank you. Okay, again, a few questions coming in, but I'll come back during the Q&A session later. Thank you. Okay, so we're a little bit over time, so we can move on. For the next talk, we have Dr. Marwa Hassan, who is a research fellow in microbiology and molecular biology at the University of Surrey. And Marwa has over 10 years of experience in diagnostics and antimicrobial resistance, with particular interest in single-cell microbiology, bacterial persistence, and in vitro and ex vivo animal models and biofilm infections. The title of her talk today is The Applications of LAMP Diagnostics for the Detection of Targeted AMR Biomarkers. So you're very welcome, Marwa. Very nice to see you, and we're ready when you are.
Thanks, Georges, and thanks for the kind introduction, and thanks for inviting me today to present. So yes, I'm a research fellow at the School of Veterinary Medicine, University of Surrey, and I'll be talking today about one of our projects that I've been working on in my current role. So antimicrobial resistance, of course, we've been talking about it in this session. So, and we are all very familiar with this figure, and we do know that even with the current pandemic, that even though this figure was a bit scary when it came out, now we know that actually the reality is probably much more worse, and that we are currently in a silent pandemic and just a matter of time until we catch up. We do also know that antimicrobial resistance does not affect only humans, and with the One Health concept that it actually affects the environment and the animals before it even considers the humans. So it's kind of how we have to always think about it from the overall perspective of the antimicrobial resistance, either on pathogens or in genes being transferred, usually across the three components. And the problem is that when we come to diagnostics or detection of antimicrobial resistance, either genes or pathogens, we always consider the standard gold method as culture. And even though I'm not against culture, and culture is great for some purposes, but it's not known for its speed or sensitivity in some cases. And when we develop molecular assays, it's always dependent on some culture-based step on enrichment to be able to remain its sensitivity and selectivity, which kind of go back to the speed of detection, which kind of affects back the concept of antimicrobial resistance with increasing prescription, because we cannot, by the time we know what resistance we have, it will be a bit too late. So we just go with empirical prescription. So and we end up in that kind of loop. So today I'll be talking about one of the projects that I've been working on, one of our One Health AJP projects, which is WorldCom. And WorldCom in particular is talking about the development of new tools for real-time detection of both zoonotic pathogens and antimicrobial resistance. And we're kind of trying to cover it from the three angles, from veterinary, human, and environmental sources. And we're using for that loop-mediated isothermal amplification, which we call it LAMP. I'm not going to go in details about how we design LAMP, but there's a very small, to the point focused mini review here if anyone is interested to look at. But basically LAMP is quite useful, but it can be easily implemented on site. So at any outbreak, it doesn't need much facilities to perform LAMP on site. It can be developed for various targets, so parasites, viruses, bacteria, or even in forensic applications. And detection signal can be quite simple once it's optimized, that it can be a color change, so from pink to yellow, or a nice amplification peak that we are all familiar with our qPCR results. Can also be on a lateral flow if need be, but usually the colorimetric one is quite more interesting because it's quite simple to analyze, especially on site when there's limited capabilities and experience. So today I'll be focused on the environmental sources, so detection from environmental sources, and I will be covering briefly how we're going on about the veterinary samples as well. So in terms of antimicrobial resistance markers that we decided to think about, so we are all familiar with the WHO critical list, with the top list full of carbaphinamases and Enterobacteriaceae. So we decided to use comparative genomics using the publicly available sequences on the database to identify the most prevalent antimicrobial resistance genes. And we particularly focused on the PLA genes, which encodes for the beta-lactamases, and the colistin, because it's one of the last resort antibiotics for gram-negatives, so MCR genes. And 4 particular pathogens of importance, both on veterinary and the human sides, so E. coli, Klebsiella, Salmonella, and Acinetobacter. And to not bombard you with a lot of comparative genomics figures, but just a quick overview or a snapshot of kind of the results. So if we look at, for example, OXA48 and 48 alleles, we do know that they are prevalent or species specific to Klebsiella, but we also find that some of the publicly deposited sequences have shared resistance with E. coli. So either E. coli acquired OXA48 or got transferred to the E. coli. Similarly, if we look at MCR1, we do know it's a species specific to E. coli. Yeah, we see some sequences that have MCR1 resistances, either Salmonella and Klebsiella. So it's kind of, even though we do know there is some resistances that are species specific, yet some other species can still acquire these resistances, which is just a matter of time until it gets spread even further. So from those figures, we kind of selected what our LAMP assays would target. So we have one assay that targets OXA48 and 48 alleles. So 181, 232, 484, and so on. The second assay, OXA23, which is currently only like predominant for Acinetobacter. And the third assay is PLA-KPC, VIM is the fourth, and MCR1. Finally validating MCR9, but today I'll be talking about the first 5. So in terms of LAMP validations, so started with using genomic DNA. So testing against a big panel of pathogens, and that is just a shortened list. So anything with a dotted line is an active control. A solid line means it's a positive sample. So 2 strains that were MCR1 positive, you can see that they were successfully amplified within two minutes. And for these, I used fluorescent-based LAMP just for quick detection and comparison of amplification time. And the rest of the negative strains, they were all negative, which was great. Similarly, if we look at the OXA48 and 48-like alleles LAMP, they were all amplified within 10 minutes with some difference in specificity, but they were still very quick, and none of the other detected, none of the other samples were positive, so that was great. And you can kind of get the idea that after the validations that the same OXA23, again, within less than 4 minutes, we can detect OXA23 and the rest were negative. And in terms of detection limits, so the dilution series and the dotted line is just a replicate. So if you can look at the last solid line, which is the blue here, so the detection limit is 0.0625, which is great, picogram of DNA, which is great in terms of detection limit. And that kind of one of the advantages of using LAMP is that it's too sensitive, so it can go for a lower detection limit. So to keep things more interesting, so we wanted to develop these or to implement these LAMP assays for detection from water samples as an example of environmental samples. So used tap water samples, and it started with 10 mils as a volume, and then developed a quick sample prep method that takes less than 1 hour to reach your LAMP results. So in order to process the water samples from the beginning into a detection or a colorimetric signal within an hour, to facilitate detection of on-site or detection from wastewater treatment plants. So in terms of the assay performance, so we spiked tap water samples with cultures, with bacterial culture, and that is to keep things more challenging, because why not? So looking at this figure, so each LAMP assay is a solid line with a different color, and then the 2 dotted lines are positive controls, and the black dotted line is a negative control. So if we look at this figure kind of here, you can see the 2 positive controls are fine, negative control is fine, and that was a water sample that was spiked with Klebsiella pneumoniae that was KPC positive, and the only positive line is a purple, which is the KPC LAMP, so that was fine. Similarly, if we look at a different spiked sample, so that was spiked with an E. coli that is post KPC and OXA48 positive, and those are the 2 positive lines that showed. So that kind of demonstrated that we can detect from spiked tap water samples directly from cultured bacterial cells without needing for culture or any enrichment steps. So in terms of detection limit, so we did detection limit by spiking bacterial cells. So the dilution series up to 10 CFU per ml. And again, the dotted lines are replicates and the solid lines are just the original. So the last line you can see is red here, and that is less than 6 minutes, and that is a 10 CFU per ml, which is quite great in terms of detection limit for detection directly from cells. So to keep things more challenging, because why not? We got some pond water. We have a pond water in front of our building. So we went on a field trip just in front of the building to get some pond water and to spike them with some bacterial cells to demonstrate that we can still detect from pond water or see if we need to modify our methods. So similar to before, solid lines are the LAMP assays and the positive controls are in the 2 dotted lines and the gray is negative. So the unspiked pond water didn't have any of the antimicrobial resistant genes, which was quite for the wildlife that we have outside, and only the positive controls that showed. And the sample that was spiked was E. coli, that is KPC and OXA48, or actually the 2 LAMP assays that actually showed positive. And we confirmed these results with qPCR. So that was actually kind of great. So I hope I showed you what we've been working on in terms of pond water. So to keep it more user-friendly in terms of going on-site for on-site testing, so we kind of transferred this platform into a colorimetric detection. So similarly, each tube got a different LAMP assay. So in order, MCR1, number 2 KPC, 48, OXA 23, VIM, and 2 positive controls and negative control. So you can guess that the first one is the unspiked, because only the positive controls that showed, and then the sample that had KPC and OXA 48 bacterial species are the tube 2 and 3 that were positive, and similarly the rest. So it kind of was great because now you can actually do that kind of assay colorimetrically offsite with easy to interpret results, and you don't need sophisticated equipment. So in terms of the second part about what we did with the detection from veterinary samples. So in collaboration with Dr. Owen Higgins, we kind of been working on using a quick sample prep method from pig feces to be able to detect against spiked or contained antimicrobial resistant genes with a streamlined straight into a colorimetric signal, so it can be also used on site, on farms, or in any outbreaks. With the idea is that the data can also be transferred through a machine learning or a smart app that has been designed by Dr. Brian Gardner at our school. So the data can be saved remotely or shared with a central lab without needing to actually having to copy or take pictures or process it on site. And that been working great. So we just kind of in the step of finally, we're just putting that all together. So just in conclusion, so I hope I showed you today that we utilize comparative genomics for the design of diagnostics tests, and that could be a useful tool. We also developed and validated the 5 AMR LAMP assays. We do have a 6-1 that is currently underway. And we also developed the rapid and sensitive water sample prep method that could be used for integrated culture-independent detection of pathogens or antimicrobial resistant genes. And lastly, that kind of detection from pond water samples was shown that it was successful within 1 hour. And lastly, we validated that we could use our developed assays for the detection of antimicrobial resistance biomarkers from fecal samples. So in terms of future prospects or plans, so we just finalizing the optimization of using the smart app so we can do the testing on site or in any remote locations. And then expanding the projects to other samples so we can kind of apply a concept of One Health diagnostics where we can detect from as many samples as possible with minimal sample prep, and that can still be extended to a wider antimicrobial resistance panel. And by that, I'd like to acknowledge my line manager, Professor Roberto Laragioni, Dr. Arno and Brian, and all our University of Surrey team. And of course, Professor Terry Smith, Deborah, Louise, Owen, Liam, and everyone at the University of Galway, and all World Health partners and One Health AGP for funding. So thank you for attention and for your time, and happy to take questions at the panel.
Marwa, thank you so much. Fantastic work and very translational. I'm looking forward to the app.
Thank you.
When it's developed. Perfect. Thank you so much. So we can move on. So for the next talk, we have with us Alexandra Swery. Alexandra, she is a research assistant in the discipline of bacteriology at the School of Medicine at the University of Galway. And she's experienced in molecular biology and holds a master's in biomedical science from the University of Galway. As a member of the antimicrobial resistance and microbial ecology groups, she has contributed to projects such as One Health EJP, WorldCom, and PEER, which stands for Public Health Impact of Exposure to Antibiotic Resistance in Recreational Waters. I hope I said that right. Her research interests include antimicrobial resistant pathogens and molecular diagnostics. We're very glad to have you here, Alexandra, and the floor is yours.
Thank you, Yorgos, for the wonderful introduction. I'm going to share my screen now. Hope you all can see this.
Yes, we can. Yeah.
Perfect. Minimize my screen there. So as Yorgos mentioned, my name is Alexandra Swery. I'm a research assistant in the discipline of bacteriology, at the University of Galway, and I'll be presenting today Peer Persistence, a longitudinal study. And Georgios so carefully told us what PEER stands for. It's Public Health Impact of Exposure to Antimicrobial Resistance in Coastal Waters. So as Bridget had mentioned in her presentation, this is now the PEER project, which had come from the ARREST project. So I'm going to talk about an overview of both projects, of PEER and the longitudinal study that we're talking about now. I'll talk about our methods, a year of communication with our participants, our results, colonization, and a summary and future plans. So everybody talked a lot about antimicrobial resistance, so I'm just going to jump right in with our study. So an overview of PEER, it began in March 2019, and we wanted to investigate whether frequent water users were more likely to be colonized in their gut with superbugs rather than non-water users. So now we have our water users, which we've named WU, and non-water users NWU. The PEER project initially started with 2-point prevalence studies, which we named PP1 and PP2. PP1 occurred during the bathing off season, and PP2 occurred during the bathing season. We compiled over 400 participants combined for both of the prevalence studies. So we sampled over 400 people, which was a great turnout, and we collected fecal samples, which was our study. Now, following point prevalence 1 and point prevalence 2, we decided to conduct a longitudinal study, which we named the persistence study, which was set to begin in October of last year, which would allow for participants from point prevalence 1 and or point prevalence 2 to be observed for colonization over the course of a year. So our persistence study began in October 2021, where we would send out sample packs every other month over the course of a year. Our first round of sample packs were sent out in November 2021, and our last round just concluded last month in October 2022. We started out with 50 participants. One was eventually excluded due to inactivity, which left us with 39 previously colonized participants. So via point prevalence 1 and point prevalence 2, that they displayed colonization with Enterobacterales. And we had 10 control participants that showed no visible colonization, also established via point prevalence 1 and point prevalence 2. We evaluated 4 methods we investigated, and 3 methods were evaluated. So to evaluate these methods, we chose 2 confirmed colonized participant samples from the point prevalence study. So these 2 participants in the point prevalence study contained multiple colonies on their plates each time. So we thought they would be good representations to test our technical evaluations, just to ensure colonies would be present. Then 3 colonies of different morphology and or color were to be selected and picked off using sterile toothpicks to be cultured on clad plates for MALDI-TOF identification. So I'll take you into our first method that we evaluated, adapted from Van Duerken 2018 and Oslam Bulkhead 2013. This involved the feces directly streaked onto selective chromogenic agar. For the purposes of our evaluation, we just use ESBL brilliance agar. This method is the one that we used for the PEER point prevalence study. So this is what we had been doing for the past 2 years and wanted to see if it was good enough for our persistence study, if it was the most valuable way to get colonies for us. So it was evaluated again, where we streaked the feces directly and incubated for 48 hours at 37 Celsius. And as you can see from the photo here, colonies of both different morphology and color result from this method. So here you can see some blue, some purple, some pink, some cream looking colors. So using this method, we were able to get 4 or 5 different colonies from this method. We also evaluated a second method adapted from Van den Bund 2020. This involved culturing 500 milligrams of feces in LB broth and supplemented with 1 milligram per liter of cefotaxime and incubated overnight at 37 C. For this particular method, we actually evaluated 2 different aliquots. So we used 20 microliters and 100 microliters. We dispensed that onto the ESBL agar plates. and inoculated using spread plate, incubated for 48 hours, like our previous method, at 37C. As you can see from the pictures displayed, colonies that resulted from this method were mostly lawn growth. There are a few individual colonies, but as you can see, they're all blue. So compared to the first method of 4 or 5 different colors, this one wasn't up to par for us. We also had method 3, which was adapted from Valdiver 2004, which involved the suspension and homogenization of a fecal swab in 5 mils of saline. The solution was vortex for 10 seconds to ensure that the feces was released from the swab. And again, we did different aliquots, 20 microliters and 100 microliters, which were dispensed to the agar plates, inoculated, and incubated for 48 hours at 37C. Now, although this looks like a lot of colonies, they are only of one type, one morphology, and one color. So you're not seeing the blues and the pinks and the purples and the creams like we saw in method 1. So still not up to the bar where we wanted it. So in the end, we didn't end up going with method 4. We considered it, it wasn't executed. This one was adapted from Royden 2019, and this involved the preparing of a homogenated feces in 1 mil BHI with 5% glycerol. 500 microliters of this homogenate would be enriched in buffered peptone water and incubated overnight. Now, we decided that it wasn't feasible for this method or practical to be completed in a timely manner, needed for the persistence study because we had a fast turnaround. So streaking the feces directly onto the plate seemed like the most practical and feasible method for our study. Now, I want to talk a bit about our year of communication with our 50 participants, now down to 49 over the course of a year. So as I had mentioned, we had sent out sample packs every other month over the course of the year. Our sample packs contained a consent form in the first pack, which all of our participants signed and sent back to us. A questionnaire, which was included in every pack, and that included questions like, have you taken antibiotics since the last time you submitted a sample? Have you traveled outside of Ireland in the past time you've given us your sample? So questions that just kept us up with our participants' lifestyles. A privacy envelope, sample collection instructions, so how to obtain the fecal sample, a sample pot and container, and a return mailer. As I had mentioned, we sent out 50 packs initially, but after our January-February samples, 49 packs were sent out. And here's our return rate at 12 months. So you can see from November to October of this year. What I want to demonstrate here is that our return rate was pretty consistent through November to June and kind of dropped off July and August and September and October. We have a few hypotheses to why that occurred. We think it's because restrictions were lifted with COVID and everyone went traveling and maybe weren't home to receive their packs and weren't able to give them back to us, send them back to us. So still a really good return rate, as you can see from this slide. In all percentages, you can see from months 1 through 8, our return rate was upwards of 90% and dropped off a little in the 9th, 10th, 11th, and 12th months. But overall, our average was 90% return rate, and that we consider a massive success, keeping in that much communication with 50 people over the course of a year. Took a lot of emails and a lot of communication on my part to keep them intrigued in our study, and we're very grateful to the people that stuck with us over the course of a year. Now I'm going to get into our results and colonization. So this graph is just going to show you from month to month how many people were colonized versus people who were undetected. I say undetected because at this point, we're unsure whether these people are not colonized at all, or if we're just not picking it up, which I'll talk about further in the next slides. But this bar graph here is going to show you month to month how many people were colonized. Now, that doesn't mean that the people colonized in November and December were definitely colonized in January and February. Some people may drop off, some people may be displaying colonization in January and February that weren't in November, December, and I'm going to show that in the next few slides, and you'll see an overview of that. So we started out with 49 participants, 39 previously colonized participants, as I mentioned earlier, and 19 of these participants were colonized with Enterobacterales at least once over the past 12 months. And we also had our control participants, which I had mentioned, and 3 of these participants were actually colonized with Enterobacterales over the 12 months, at least once, which is a big change from not being colonized in point prevalence 1 or point prevalence 2, as I had mentioned. That's why we picked them as control participants. Somewhere along the way, they did display colonization, which was interesting to us. Now, if you do a quick maths, that adds up to 22 persistent participants colonized with Enterobacterales at least one time over the past 12 months. 7 of these were water users, and 2 of these were non-water user controls. I'm sorry. We also had 12 non-water users and 1 non-water user control from this group. Now, here is a big handy-dandy graph of all of our persistence participants, non-water users that showed colonization over the past 12 months. You can see point prevalence 1 and point prevalence 2 labeled in the beginning, and then 1 through 6 are sampling rounds. So as I mentioned, every other month we sent them a pack, so 6 packs in total from our participants. Now, red represents Enterobacterales growth and green would be no growth. Yellow are our controls, and gray, we received no pack back from them, unfortunately. So we had 13 non-water users who were colonized over the course, at least once over the course of 12 months. I want to draw your attention to these 2 in particular because they were our special ones. They were colonized every single sampling round over the past 12 months. We also want to draw your attention to participant number 11, who was one of our participants that was seemingly colonized and then seemingly not colonized from month to month. This will come into play when we do sequencing further down the line to investigate whether the Enterobacterales were there and not picked up, or are they really going from month to month, picking something up and then dropping something and then picking something up again, which we thought was very interesting. For our water users, we only had 1 participant who was colonized all 6 rounds, I'm sorry. And this participant did not participate in the first point prevalence 1, but did participate in point prevalence 2. So colonized every single time we sampled them, which was very interesting to us. Altogether, we had 23 participants that presented with at least 1 Enterobacterales over the last 12 months. I'm just going to rapid fire some bacteria at you. So we had 17 participants presenting with E. coli, 1 with a Klebsiella aerogenes, 6 with Streptobacter frondae, 1 with a Rotella ornithinolytica, and then we had 3 participants presenting with 2 different Enterobacter species, 2 participants presenting with 2 different Serratia species, and 1 participant presenting with a Hafnia. Now, if you do a quick maths, that doesn't add up to 23, but this is because 5 participants presented with multiple Enterobacterales. So it could be that in one sampling round, a participant presented with several different colonies, one of which was an E. coli, another of which was an Enterobacter, another of which was a Klebsiella. So that's why it doesn't add up to 23, but also good for us, bad for the participant, as we like to say. Now, altogether, we presented with 130 individual Enterobacterales isolates over the last 12 months. 104 were phenotypic ESBL isolates, 21 were phenotypic AMP-C isolates, 4 were CRE isolates, and 8 were non-ESBL, AMP-C, or CRE isolates. So 8 of these isolates, although they presented as they grew on ESBL brilliance agar and M supercarba agar, grew on our selective agars. When we did AST, they ended up being completely susceptible to our antibiotics. So we considered them non-ESBL MC or CRE. Now, one isolate showed characteristics of an ESBL and a CRE. One isolate showed characteristics of an ESBL and AMP-C. Two isolates presented with characteristics of AMP-C and CRE. And one lucky participant, an isolate showed characteristics of an ESBL, AMP-C, and a CRE. So because this is still an ongoing project, I have a bit of a summary for you. And our future plans. So our recruitment we considered very successful with an average of 90% return rate over 12 months. And as for our colonization, roughly 45% of our persistence participants displayed Enterobacteriales colonization at one point or another. Our future plans, I had mentioned a few of them already. So we're doing short read sequencing currently, and metagenomics on DNA extracted from fecal samples provided by participants is another thing in the works. That'll be very useful for participants, like I had mentioned, that seemingly dropped off and then picked up some Enterobacteriales along the way. So whether they were colonized the entire time and our agar plates just weren't picking them up, or are they really going from 1 month to the next colonized and not colonized? And after our short read sequencing, we'll be doing in-house long read sequencing after our short reads are analyzed. I'd like to thank you all for your attention, and I'd like to thank the EPA for funding us, the peer team, Dervla, Liam, Maeve, Louise, Sinead, and Dimitri, and all the members of the ARME group for their continued feedback and support. And my LinkedIn QR code is there if you'd like to reach out to me or learn more about the project. Thank you.
Thank you so much, Alex. Thank you. Fantastic work. And I must say, as a swimmer, I'm a bit relieved. After what we did presented, I'm a bit relieved as to what you presented now, so I can go back to swim. Perfect. So we have a few questions. I may take them in order of presenter. So there's a few questions for each one of you. If any of the attendees have more questions, just feel free to put them in the Q&A chat box, and I will then relay them on to the presenters. Okay, so Bridget, thank you very much, first of all, for the keynote. Very, very interesting. A lot of work into it.
Absolutely. Thanks so much.
So first of all, my question is, how easy was to collect all the samples over the years?
Yeah, that's a good question. A lot of mileage on the road, a lot of carrying heavy water samples. So, you know, absolutely the results shown are, you know, kind of a very brief summary, but, you know, I think like in terms of the distance we've traveled to collect the samples, the hours in the laboratory, all of that side of things, you don't see, you know, with presentations. So although it was a lot of work, it was very enjoyable nonetheless. It was brilliant to meet people in different laboratories, you know, to actually go out into the environment and have that part of your day job as well was really lovely. So yeah, in terms of sample collection and processing, there was a lot of work, but very enjoyable.
Brilliant, brilliant. So the next question, it relates really to the genes you identified in terms of carbapenem resistance. You mentioned, I think, OXA48 as being one of the most prevalent one. So the question is, how does it relate to actual clinical settings in Ireland? Is in terms of carbapenemases, is OXA48 something that predominates clinics as well, or you know, how does this link with clinical environments?
Yeah, so absolutely. So we definitely, from looking at the reports from the CPE reference lab, we do see that OXA48 is the most common type, and particularly from the data in 2020 that was published, we can see that the majority of the isolates were OXA48 producers. But again, I suppose, luckily, we don't see that OX48 confers a high level of resistance to meropenem. It's more intermediate. Now, again, that does have consequences in terms of treatment. But yeah, I suppose the types that we did find do match up with those in circulation in the clinical environment. Again, another, I suppose, potential hint that the isolates in the environment may come from, you know, human sources.
That's worrying,
Yeah.
to say the least. And you've mentioned, how many locations are there in Ireland currently where there's untreated sewage entering the water directly?
So as of 2020, there were, I must just double check myself to be sure, but as of 2020, there's 34 locations. So I haven't checked the most recent reports, but I did, I suppose, report on the numbers that were present during the sample collection process. So they are working to reduce this number by putting in, you know, new wastewater treatment plants and redirecting discharges that are present to current treatment plants. So there is plans to eliminate those over the next couple of years, but still a significant number around the coast of Ireland.
Okay. So the next question I was thinking is, so I suppose it's more of a technical note, really, because the way you applied these methods, you're really picking up, I suppose I'm comparing culture-dependent, culture-independent methods here, but the way you applied it, you're making sure, instead of using metagenomics, you're making sure that whatever you're seeing is actually alive, which is very important. So this, whatever you identified in the water was not just lysed DNA of enterobacteriales that entered through sewage. These bacteria were actually there, they were metabolically active and alive. You recovered them. So I wonder how exactly you did that. What's the method that you used to extract the water and so on?
Yeah, that's a good question. So for our water samples, we filter 30 litres of water using, I suppose, a small filter that will kind of pick up and capture any bacteria that are present. We then enriched it using buffered peptone water and then plated it directly onto different chromogenic agars. So that allows us, as you said, to actually recover viable bacteria that were alive. For our sewage samples then, I suppose similarly to the PEER project, we did a direct plating technique where we just, actually used a swab and dipped it in our sewage collected and again, directly plated it onto our agar plates. So I think you could have a whole session on, the comparing genomic versus culture-based methods. There's definitely pros and cons for both, but I don't want to take up any more time anyway today.
No, that's very good. I think you need both. That's I think they're complementing each other. So you certainly need both, because I suppose the next question is as to into what quantity these enterobacterial drug-resistant enterobacterials are present and whether they're able to colonize, as Alex showed later, or whether they're not able to colonize and whether they're able to cause infection or not cause infection. So,
exactly. That's definitely the next piece of the puzzle.
Yeah. Perfect. Thank you so much, Bridget.
Perfect. Thanks, Georgios.
So the second speaker was Francesca. Hi, Francesca.
Hi.
So a few questions for you that they have come in. One of them was from the, first of all, very interesting link there. So we're seeing patients that they're on clozapine, that their treatment options are very limited. If they get an infection, that they get disproportionately affected with pulmonary infections, particularly pneumonias. And then you identified a colonization rate of, what was 51%?
51, yeah.
Which compares to
the healthy population, it would be 5%, and then immunocompromised is 60. So it's quite high.
So clearly something's going on with the oral microbiome of these patients. So the question was, from the species you identified, most of them were Klebsiella oxydoca, if you remember. So is there any hypothesis as to why most of them were Klebsiella oxydoca, particularly given that these patients are coming in every month to the clinic?
Yeah, I guess Klebsiella is quite, it's one of the biggest pathogens from like Enterobacterialis altogether. But Klebsiella, including Klebsiella oxydoca, is known to be able to survive for very, very long times on surfaces. And Klebsiella oxydoca in particular is known to, it's becoming emerging more and more infections in community or health-associated infections, particularly those in patients that are immunocompromised or patients that are in critical care facilities in the hospital. It's a growing number, so that could possibly be where this high rate of Klebsiella oxydoca is coming from.
Okay, That's very interesting. And then you also saw an association there with some patients being colonized with very similar strains. Is that correct? Yeah. And so I suppose in terms of hypervirulence, what exactly did you note in your genomic data?
In particular with the isolates that are shown to be highly virulent, there was a presence of the Yersinibactin operon, the Aerobacter operon as well. So that was present in several of the isolates. So it would be interesting maybe to do more analysis. That's ongoing at the moment, is what virulence factors were in what isolates. So interesting to see that, not only were these pathogens in the mouths of these patients, but they were also like highly virulent, but none of the patients were in an active infection. They were just colonized with the pathogens.
And I suppose the key question then would be whether they're persistently colonized or this strains.
So yeah, that would be ideally when we went first to recruit the patients, we did include in the consent form if they would be willing to participate in future studies. So quite a majority of the patients did agree, and they said that they would. So one part of future studies would be to go back and to take samples from these patients again and to determine if it was a transient or persistent colonization that we did observe.
Perfect. And you did mention also there are plans for some neuromicrobiology association as well.
Yeah, to try and associate back into the etiology of schizophrenia and determine, is the oral dysbiosis as a result of, is it the drugs that they're using? Is it as a result of the disorder itself? Is it contributing to the symptomatology that's seen? And possibly, is there a difference in the oral microbiome dysbiosis seen in patients that have schizophrenia versus patients that would have treatment-resistant schizophrenia? So it'd be interesting to see if there is a direct link there between the pathogens and the neurological signaling.
Okay. And you did mention also Calamniella and one new species as well of Calamniella, is that right? And then there's the association between Calamniella and International Space Station. So one of the questions that came into the Q&A is, were the Calamniella isolates from the same patient? The two ones that they're the Pearsonii and the other one that it's a different Calamniella?
No, they weren't. They're from two different patients. And they weren't from the same day either. They were from later on. I think there's maybe about a week or two between the two patients.
Okay, that's very interesting. I suppose you haven't named the new species yet.
No, early days yet.
Perfect. Okay. Thank you so much. Thank you so much, Francesca.
Thank you.
And then Marwa, thank you very much. Very, very interesting talk. And as I said, I'm very much looking forward to the app. Very translational as well. Excellent work. A few questions for you coming in. So one of them says, very interesting research. Is this assay something you think would be included in clinical and environmental analysis also for fast results?
That's a very good question. I think maybe before the pandemic, the answer to that question was a bit different from after the pandemic, and definitely COVID increased the conversation over IP and having a quick assay that could be on site for even at least quick screening. So I think there are quite few IPs that should be out soon to have some LAMP-based assays for quick screening at least, so it can be saved or used for any outbreaks.
Okay, perfect. And one question I had was, so you did mention MCR, which is highly worrying because it confers resistance to colistin, and which is the last of last resorts we have left in clinical practice. So how much of MCR you've actually, when you're testing samples from water, and you've mentioned PIDs as well, how much of that have you seen around?
Because the concept of what we've been doing was mainly for development, for the development stage. So for the samples that we've tested, we haven't seen colistin resistance in them. So because they were negative, we had to spike it from the outside. But for the comparative genomics work, yeah, there were a lot of MCR ones. So yeah.
Okay. Well, worrying again. So the next question is in the Q&A as well. So you've mentioned that the limit of detection is about 10 CFUs. Is that as a limit of detection, is that considered high, or as you might be missing some positive samples, which could be less than 10 CFUs?
So that is 10 CFUs for the 10 mLs of water that we processed.
Oh wow, okay.
So that means it's a roughly 1 CFU per ml of water. So of course, if you increase the water sample, you might actually get higher or lower detection limit. And to be honest, I haven't gone in this dilution series lower than 10 CFUs per ml because I thought it's not going to get lower than that. But then when it worked, I was like, okay, I'm not going to repeat that either. So, so if we do another dilution series going lower than 10 CFUs per ml, it might detect even lower than that.
Perfect. Okay, that's very, very impressive. And the next question I had was, and that may be a silly question, but so you've mentioned you tested water, you've mentioned you tested pigs as well. Is there any plans, are there any plans to check human fecal matter as well, or is that something that is beyond?
Not within the current scope, because our project is ending end of this year, but it's certainly something of interest that to kind of expand on. But what we're expanding on at the moment is detection from urine, so detection of UTI infections. So that kind of could work with humans for obvious reasons, easier and quicker than fecal samples.
Okay, perfect. Again, fantastic work. Fantastic work. Indeed. Thank you so much, Barbara.
Thank you for having me.
Okay, Alex. Few questions have come in for you as well. One of them was, so having managed ongoing engagement with so many participants, what would you recommend to future projects to firstly recruit and then encourage ongoing engagement from participants? Great presentation, very interesting.
Oh, thank you. That's very sweet. So yeah, I think that we as scientists tend to forget that participants are people a lot of the time, and what they want is interaction. So the way I kind of communicated with them is I would send out an email when we were sending out the packs, and then a week later a reminder email. And occasionally, if we didn't get enough packs back, I would send another email. I'm sure they were sick of hearing from me a lot of the time, but send another email reminder and just said, if you need another pack, let us know. If you'd like to withdraw from the study, let us know. You know, treat them like you're chatting to them, like you're talking to them. And I think recruiting through the pandemic was in some ways beneficial to us, even though it didn't seem like a blessing at the time. But we did a lot of recruiting online through newspapers and through social media. And I think during the pandemic, a lot of people were looking to get involved in something else, which was to our benefit, I guess. And then those people that we recruited wanted to stay on and continue with us because we had established that relationship with them in the beginning. So I think it's all about establishing relationships with the participants. Now, not to get personal with them or anything, but to treat them like people. I think we tend to forget a lot of the times when you're dealing with their samples. It's nice to separate the people from the samples a lot of the time, but we do have to, be reminded of that occasionally.
Absolutely, absolutely. What a great point to make. Absolutely. And okay, one of the other questions was, so is there any hypothesis as to explain this discrepancy that you observed between streamers and non-streamers in terms of colonization?
So a hypothesis at the time that we have, and continuing with the point prevalence study of the PEER project, we think that our hypothesis is that a possible contributor to, water users not being as seemingly colonized as non-water users is that water users are just generally more active, more healthy. They could be taking better care of their diet, better care of their body, and aren't as susceptible to these bugs. Maybe they're just flushing them out quicker than non-water users are, who don't frequently swim or are active in the water. So that's kind of our leading hypothesis now. We did take in a lot of data about their diet and about how often they do swim and whether they're healthcare workers, whether they're have animals or pets at home, whether they're farmers. So we do have all of that data. So hopefully with the persistence study, we'll be able to sort through it more and see if there are any connections in combining our point prevalence study and our persistence study. So leading hypothesis is that might be healthier, might not be.
Fair enough. And I think overall, though, it's a very positive message, and we do need that. We need sometimes, I mean, people swimming, it's such a wonderful recreational activity, particularly when you have coastal, well, I was going to say when you have coastal waters like the one we have in Ireland, but apart from particular locations, let's say, but overall, the Irish coastal waters are of excellent quality. So it's such a wonderful activity to have. So it's a very positive message, which we rarely see in research. We normally are dominated by worrying and negative outputs. So it's great to have that. It's very good.
We'll be going swimming soon enough.
And you did also mention you have some plans for culture independent work, because I think that's highly relevant here. Try to understand why these people are colonized certainly has something to do with their microbiome. So you did mention you have some plans for that as well, if I'm correct.
Yes, or we're planning on doing a metagenomic study where we take our fecal samples that we've collected over the past year and we extract the DNA from that, and then discover kind of if we're picking up everything that was in that fecal sample or not. It's possible that our selective agars didn't pick up everything. Culture work isn't an exact science, I guess. You know, there are bugs that tend to hide occasionally. So our metagenomics work will definitely show whether or not these people are colonized consistently or if we're just not picking it up. And I think that'll be really interesting to see, especially for our participants who maybe were colonized the first sampling round, but then weren't colonized until round 6, which was our last sampling round. So were they colonized that entire time, or did they have something in last November that they picked up again this time this year?
I'm very much looking forward to this result. Perfect. We can leave it here. I think we're a little bit over time. Thank you so much, everyone, for presenting today. And I want to apologize to the attendees again for the technical difficulties we had in the beginning. We're way over them now. Everything's running smoothly. So in terms of tomorrow, we're back at 9 o'clock. You can join using the same link that you used today. We're starting with a focus on emerging pollutants session chaired by Dr. Leah Morrison. And then we have the focus on people, nature, environment that is chaired by Dr. Alexandra de Menezes. And lastly, we are closing with the behavioral societal change session chaired by Dr. Sonia Duane. And Dr. Leah Morrison then will have the closing session as well at 12:45 tomorrow. So thank you all so much for presenting today. Thank you very much to the chairs. Apart from myself, of course, to all the other chairs as well, to Professor Morris, Dr. Carolina Verissimo, and thank you all for attending. And we're very much looking forward to seeing you all tomorrow. Thank you so much.